From the very dawn of human existence, etched into the earliest records of our ancestors, lies a fundamental, almost primal impulse: the desire to heal, to alleviate suffering, and to comprehend the mysteries of the human body and its afflictions. This deep-seated quest, born of vulnerability and a profound will to survive, forms the very bedrock of what we now call medicine. Our journey through the evolution of modern medicine is not merely a chronicle of scientific breakthroughs, but a sweeping epic of human ingenuity, perseverance, and an unending intellectual curiosity. It is a testament to our capacity to observe, to question, to experiment, and to continually refine our understanding of life itself. Imagine our earliest forebears, confronted by a raging fever, a broken bone, or an inexplicable illness. Without the tools of science, their world was one of potent forces, spirits, divine favor, or malevolent curses governing health and disease. Healing, then, was intertwined with magic, ritual, and a nascent understanding of natural remedies. Yet, even in these primordial beginnings, we see the nascent threads of empirical observation. The knowledge passed down about which plants eased pain, which poultices staunched bleeding, or which practices brought comfort. This era, shrouded in the mists of time, represents the initial, crucial steps of medicine's timeless quest, a quest driven by an urgent need for practical solutions, even if the underlying mechanisms remained entirely unknown. As civilizations blossomed, so too did more structured approaches to health. From the sophisticated medical papyri of ancient Egypt to the meticulously codified laws of Mesopotamia, we witness the gradual emergence of specialized healers, surgical instruments, and even rudimentary public health concepts. But it was in ancient Greece, with figures like Hippocrates, that a revolutionary paradigm shift began to take root. The radical idea that disease was a natural phenomenon, with natural causes, rather than divine punishment, marked a watershed moment. This emphasis on rational observation, detailed clinical notes, and the pursuit of ethical conduct laid the philosophical and methodological groundwork that would resonate through millennia, forever shaping the very identity of the physician. The Roman Empire, while inheriting much from Greek thought, championed practicality and public health on an unprecedented scale. Their enduring legacy of aqueducts, sewage systems, and military hospitals demonstrated a profound understanding of environmental factors in health and the importance of organized care. Following the decline of Rome, much of classical medical knowledge found sanctuary and further development in the Islamic Golden Age. Brilliant scholars and physicians like Avicenna and Rhazes not only preserved and translated ancient texts, but expanded upon them with meticulous clinical observation, advancements in pharmacology, and innovative surgical techniques, acting as vital bridges between ancient wisdom and future enlightenment. The Renaissance in Europe, a period of fervent intellectual rebirth, ignited a renewed passion for anatomical study. Andreas Vesalius, with his groundbreaking dissections and meticulous illustrations, dared to challenge centuries of Galenic dogma, correcting long-held misconceptions and ushering in an age of anatomical accuracy. This spirit of inquiry, fueled by the invention of the printing press, democratized knowledge and catalyzed further revolutions. William Harvey's astonishing discovery of blood circulation, for instance, shattered previous models of physiology and paved the way for a mechanistic understanding of the human body. These were not mere discoveries. They were fundamental reimaginings of what it meant to be alive and how the body functioned. The journey then accelerated into the scientific revolution and the age of enlightenment. The microscope, a seemingly simple invention, unveiled an entire unseen world of microorganisms, hinting at a new dimension of disease. Clinical observation became more systematic, diseases were classified, and the foundations of pathology were meticulously laid. Yet, despite these advances, the true nature of many diseases remained elusive, and devastating epidemics continued to plague humanity, underscoring the formidable challenges that still lay ahead. The 19th and 20th centuries witnessed an explosion of understanding, transforming medicine from an art of observation and crude intervention into a rigorous, evidence-based science. The germ theory of disease, pioneered by titans like Louis Pasteur and Robert Koch, unequivocally linked microscopic organisms to specific illnesses, unraveling the enigma of infection and paving the way for antiseptic surgery, vaccinations, and the eventual dawn of antibiotics. These were truly transformative moments, reducing mortality rates on a scale previously unimaginable and extending human lifespans dramatically. The discovery of anesthesia revolutionized surgery, turning agonizing procedures into manageable interventions, while the advent of X-rays peered non-invasively inside the body for the first time. The latter half of the 20th century and the dawn of the 21st have brought forth an era of unprecedented progress. The unlocking of the DNA double helix launched the age of molecular biology, leading to the Human Genome Project, gene therapies, and the promise of personalized medicine. Organ transplantation, advanced medical imaging like MRI and CT scans, sophisticated immunological treatments, and the integration of artificial intelligence and big data are continuously reshaping diagnostics, therapeutics, and preventative care. Medicine today is a vast, interconnected web of specialized knowledge, technological prowess, and ethical considerations. Yet, amidst this breathtaking progress, medicine's timeless quest endures. While many infectious diseases have been conquered, new challenges constantly emerge. Antibiotic resistance, the rise of chronic non-communicable diseases, the complexities of autoimmune disorders, and the enduring threat of global pandemics. Furthermore, the very power of modern medicine brings with it profound ethical questions about life, death, equity, and access. This comprehensive essay will trace this remarkable evolution, charting the major turning points, introducing the visionary minds who dared to challenge prevailing wisdom, and exploring the societal forces that shaped medicine's trajectory. From the first shaman's whispered chant to the precision of genomic editing, we will witness how our understanding of the human body, disease, and healing has been continually refined. It is a story not just of science, but of humanity's persistent drive to understand itself, to overcome adversity, and to continuously improve the quality and longevity of life. Let us embark on this extraordinary journey through the evolution of modern medicine, a quest that continues to unfold before our very eyes. Before the sterile operating room, the precise diagnostic scan, or the targeted pharmaceutical, there was the wilderness. There was raw survival, constant vulnerability to injury, disease, and the unpredictable forces of nature. In this primordial landscape, medicine was born not of scientific theory, but of urgent necessity and an innate human drive to alleviate suffering and preserve life. This earliest phase of healing, stretching back tens of thousands of years, was a profound blend of deeply held spiritual beliefs and rudimentary, yet often remarkably effective, empirical observations. It was a world where illness was often seen as a spiritual imbalance, a curse, or the work of malevolent forces, yet practical solutions derived from trial and error were equally sought after. Imagine our Stone Age ancestors. A broken bone, a festering wound, a persistent fever, or the inexplicable onset of a painful illness could mean swift death. Without any understanding of anatomy, physiology, or microbiology, the human body and its ailments were terrifying mysteries. Thus, explanations were sought in the spiritual realm. Disease was attributed to evil spirits entering the body, the wrath of offended deities, the breaking of taboos, or the maleficence of a sorcerer. Consequently, healing rituals were primarily focused on spiritual intervention. Appeasing gods, expelling demons through incantations, dances, or sacrifices, and invoking the aid of benevolent spirits. Central to this early system was the figure of the shaman, the witch doctor, or the medicine man, individuals often perceived as possessing a unique connection to the spirit world. These healers were not merely spiritual guides. They were also keen observers of their environment. They accumulated vast traditional knowledge about the properties of plants, minerals, and animal parts, knowledge passed down through generations. They understood which leaves could staunch bleeding, which roots might soothe pain, which bark could reduce fever, or which preparations could induce vomiting to expel perceived toxins. This practical, experiential knowledge, born of countless observations and desperate experiments, formed the empirical backbone of prehistoric medicine. Archaeological evidence provides fascinating glimpses into these practices. Skeletal remains from ancient sites around the world show healed fractures, demonstrating that early humans had skills in setting bones. Perhaps the most intriguing evidence is the widespread practice of trepanation, the surgical drilling or scraping of a hole in the skull. Found in cultures across continents and dating back thousands of years, trepanation was likely performed for various reasons, including the relief of headaches, epilepsy, or mental illness, often under the belief that it would release evil spirits trapped within the cranium. The presence of bone regrowth around these openings indicates that many individuals survived the procedure, speaking to a surprising level of surgical skill, even in rudimentary conditions. While motivated by spiritual beliefs, the execution was undoubtedly a physical, empirical act. As humanity transitioned from nomadic hunter-gatherer societies to more settled agricultural communities, and eventually to complex civilizations, medical practices became more organized, though the spiritual dimension remained profoundly influential. In ancient Mesopotamia, the cradle of civilization between the Tigris and Euphrates rivers, illness was still primarily viewed as a punishment from the gods or the work of demons. Healing rituals involved elaborate incantations, exorcisms, and divination, reading omens from animal entrails or celestial phenomena to diagnose and prognose ailments. The Ashipu, or exorcist physician, was skilled in diagnosing the divine will, while the Asu, or empirical physician, focused on herbal remedies and practical interventions. Despite the heavy reliance on divination and magic, Mesopotamian medicine also displayed a remarkable degree of empirical observation. Clay tablets from this era detail an extensive pharmacopoeia, listing hundreds of medicinal plants, minerals, and animal products used in complex prescriptions. Some tablets, like the Diagnostic Handbook from the 7th century BCE, even show a surprisingly systematic approach to diagnosis and prognosis, listing symptoms and predicting the likely course of a disease. Furthermore, the famous Code of Hammurabi circa 1754 BCE included detailed laws governing the fees and punishments for surgeons, indicating that surgical procedures likely rudimentary. But still, actual physical interventions were a recognized part of Mesopotamian medical practice, even if the underlying rationale was still tied to spiritual belief. Surgeons performed procedures such as setting broken bones, lancing abscesses, and even crude eye operations. Moving westward, ancient Egypt developed a more sophisticated and specialized medical system, arguably the most advanced of the ancient world. While Egyptian medicine, too, was steeped in religious belief, gods like Sekhmet, goddess of healing and plague, and Thoth, god of knowledge and writing, were central figures, and the deified physician Imhotep was revered. It also showcased a profound commitment to empirical observation and practical skill. Egyptian physicians were highly specialized, with titles such as guardian of the anus, dentist, or physician of the eyes, suggesting a division of labor that hints at a more systematic approach. Their knowledge of anatomy, though not based on dissection for scientific understanding, benefited significantly from the practice of mummification. While the embalmer's primary goal was preservation, their repeated handling of internal organs provided an unparalleled familiarity with their location within the body. Two key medical papyri provide invaluable insights into their medical knowledge. The Ebers Papyrus circa 1550 BCE, one of the oldest and most extensive medical texts, contains over 700 remedies, spells, and incantations for a wide array of ailments. It's a fascinating blend of practical remedies for conditions like asthma, diabetes, and eye diseases, alongside magical spells for driving out demons. In contrast, the Edwin Smith Papyrus circa 1600 BCE, believed to be a copy of a much older text, is remarkably rational and devoid of magic. It describes 48 cases of surgical trauma, detailing examination, diagnosis, prognosis, and treatment in a methodical, logical sequence that is surprisingly modern in its clinical approach. It discusses fractures, dislocations, wounds, and tumors, often recommending practical solutions like splints, sutures, and cauterization. Egyptian treatments involved a vast pharmacopoeia of herbs like castor oil, opium, senna, animal products, and minerals. They employed various diagnostic techniques like pulse taking and urine examination. Their surgical instruments were sophisticated for their time, and they understood the importance of hygiene, cleanliness, and public health measures to a degree that would not be consistently seen again for millennia. In essence, the earliest forms of healing, from the prehistoric shaman to the specialized physicians of Mesopotamia and Egypt, were characterized by a dynamic interplay. On one hand, there was a pervasive belief in supernatural causation and intervention, leading to rituals, spells, and divine appeals. On the other, there was a continuous, accumulating body of empirical knowledge derived from observation, trial and error, and practical necessity. This dual approach, where the spiritual often provided the framework and the practical offered the immediate relief, laid the essential groundwork for what would eventually evolve into the rational, scientific discipline of medicine. These early healers, driven by the timeless quest to overcome suffering, were the essential first steps on a very long and transformative journey. Following millennia, where healing was inextricably bound to magic, divine intervention, and the appeasement of angry spirits, a revolutionary intellectual current emerged in ancient Greece, ushering in what we now recognize as the birth of rational medicine. This profound shift marked a pivotal moment in human history, laying the foundational principles of observation, logic, and ethical conduct that continue to underpin modern medical practice. It was here, in the cradle of Western philosophy, that the focus began to move from supernatural forces to natural explanations for disease, fostering a systematic approach to understanding the human body and its ailments. The towering figure associated with this transformation is Hippocrates of Kos, often revered as the father of medicine. While historical records are often vague, and it's likely that many texts attributed to him were actually compiled by various authors over centuries, collectively known as the Hippocratic corpus, his school of thought profoundly influenced the development of medicine. The Hippocratic physicians were radical in their insistence that illness was not a punishment from the gods nor a demonic possession, but a natural phenomenon with observable, natural causes. This was a direct challenge to the prevailing religious and superstitious beliefs of the time. They proposed that factors like diet, environment, lifestyle, climate, and even geographical location played crucial roles in health and disease. At the heart of Hippocratic medicine lay the groundbreaking emphasis on meticulous observation. Physicians were encouraged to carefully examine their patients, noting symptoms, changes in bodily fluids, the progression of fever, and environmental factors. They were less focused on miraculous cures and more on prognosis, predicting the likely course and outcome of a disease. This required not only acute observation, but also detailed record-keeping, building a cumulative body of knowledge from individual cases. The physician's role was seen as aiding nature in its healing process, not fighting against it. This concept, known as physis, recognized the body's inherent capacity to heal itself, and the physician's duty was to create optimal conditions for this natural healing to occur. Therapeutic interventions were often gentle and supportive, including rest, appropriate diet, exercise, and bathing. Perhaps the most influential, albeit ultimately incorrect, theory that emerged from this period was the humoral theory. This theory proposed that the human body contained four fundamental fluids, or humors: blood, phlegm, yellow bile choler, and black bile melancholy. Health was believed to be a state of balance among these humors. An imbalance, or dyscrasia, led to disease. For example, an excess of yellow bile might cause fever and irritability, while an excess of black bile could lead to melancholy. Treatments, therefore, aim to restore this humoral balance through various means, such as bloodletting to reduce blood, purgatives to remove excess bile, or emetics to induce vomiting for phlegm. While scientifically flawed by modern standards, the humoral theory was a monumental leap forward. It provided a coherent, naturalistic framework for understanding disease that did not rely on supernatural explanations. It was a rational attempt to explain internal bodily processes, even if the model itself was inaccurate, and it dominated medical thought for nearly 2 millennia. The Hippocratic physicians also championed a holistic approach to patient care, recognizing that a person's well-being was not merely the absence of disease, but a complex interplay of physical, mental, and environmental factors. They considered the patient's constitution, age, occupation, and even emotional state when formulating a diagnosis and treatment plan. This comprehensive view fostered a deep respect for the individual and acknowledged the interconnectedness of various bodily systems with external influences. Their writings contain numerous insights into epidemiology, recognizing patterns of disease occurrence in relation to seasons, water sources, and prevailing winds, demonstrating a remarkably early understanding of public health principles. Beyond their contributions to clinical practice, the Hippocratic school also laid the foundations for medical ethics. The Hippocratic Oath, though its exact origins and authorship are debated, stands as a timeless declaration of ethical principles for physicians. It emphasizes patient welfare above all else, advocating for compassion, privacy, and the avoidance of harm. Phrases like primum non nocere first, do no harm, resonate throughout medical history. The oath binds the physician to treat all patients equally, without prejudice, and to maintain the confidentiality of medical information. It establishes a professional ideal, elevating the practice of medicine from a mere craft to a calling imbued with profound moral responsibility. This ethical framework has profoundly influenced medical education and practice for centuries, creating a sense of trust and professionalism that remains vital today. While the core of rational medicine stemmed from observation and non-invasive methods, a significant development in the understanding of human anatomy occurred slightly later, in the Hellenistic period, particularly at the famous Library and Museum of Alexandria in Egypt. Here, pioneers like Herophilus circa 335 to 280 BCE and Erasistratus circa 304 to 250 BCE were permitted to perform human dissections, a practice largely forbidden in mainland Greece due to religious and cultural taboos. This freedom allowed them to make unprecedented discoveries about the human body. Herophilus distinguished between arteries and veins, described the nervous system, and differentiated sensory from motor nerves, and recognized the brain as the center of intelligence. Erasistratus contributed to the understanding of the heart and its valves, and the distinction between the cerebrum and cerebellum. These anatomical insights, though not immediately integrated into widespread clinical practice in Greece, were crucial stepping stones, providing a more precise map of the body that would later be synthesized and expanded upon by figures like Galen. The legacy of ancient Greek medicine, particularly the Hippocratic tradition, is immense and enduring. By rejecting supernatural explanations, promoting rational observation, advocating for a holistic approach, and establishing a strong ethical foundation, they initiated a profound paradigm shift. They moved medicine from the realm of ritual and superstition towards a nascent science, where systematic inquiry and empirical evidence began to guide diagnosis and treatment. While their theories, like humorism, were eventually superseded by more accurate scientific understanding, their fundamental methodological approach, the rigorous observation of symptoms, the emphasis on prognosis, the belief in natural causes, and the unwavering commitment to ethical practice laid the indispensable groundwork upon which the entire edifice of modern medicine would eventually be constructed. This period represents the crucial intellectual awakening that set medicine on its path to becoming a true scientific discipline. Where ancient Greek medicine, as we've explored, excelled in philosophical inquiry, rational observation, and ethical principles, the Roman world, with its profound pragmatism and unparalleled engineering prowess, shifted the focus of medicine dramatically towards public health, infrastructure, and organized care. The Romans were builders, administrators, and conquerors, and their approach to health reflected these characteristics. It was less about abstract theories of disease and more about tangible solutions for the collective well-being of their vast empire. One of Rome's most enduring and impactful legacies lies in its monumental contributions to public health infrastructure. Unlike the Greeks, who emphasized individual health and philosophical balance, the Romans understood that the health of the individual was intricately linked to the health of the community. Their innovations in urban planning and sanitation were unprecedented and would not be matched for over a millennium. At the core of this public health revolution were the magnificent Roman aqueducts. These engineering marvels, some of which still stand today, brought fresh, clean water from distant sources directly into Roman cities. This consistent supply of potable water was transformative. It allowed for basic hygiene, provided water for public fountains, and crucially, helped prevent the spread of waterborne diseases like cholera and dysentery, which historically decimated urban populations. Consider the stark contrast. In most ancient cities, water was often drawn from contaminated local wells or rivers. Rome's vast network of aqueducts ensured a healthier populace by significantly reducing the burden of water-related illnesses. Sextus Julius Frontinus, a Roman official, even meticulously documented the city's water supply in his work, De Aqueductu Urbis Rami, detailing flow rates and quality, showcasing an administrative commitment to public health. Equally critical were Rome's sophisticated sewage systems. The most famous, the Cloaca Maxima, or Great Sewer, initially constructed in the 6th century BCE, was a colossal engineering feat that drained marshes, carried away rainwater, and critically, removed human waste from the city. This extensive network of underground drains, combined with public latrines often connected to running water, revolutionized urban sanitation. By effectively removing waste, the Romans dramatically reduced the presence of disease-carrying pests and the contamination of living spaces, thereby curtailing the spread of many infectious diseases that thrived in unsanitary conditions. While they didn't understand germ theory, their practical measures achieved similar health benefits. Beyond water and waste management, the Romans embraced public baths, thermae, as central to daily life. These grand complexes, accessible to nearly all citizens, served not only as places for personal hygiene, but also as social hubs, gymnasiums, and relaxation centers. The regular practice of bathing, combined with the use of strigils to scrape off dirt and oil, contributed significantly to personal cleanliness. While the communal nature of the baths could sometimes facilitate the spread of certain skin ailments, their overall impact on general hygiene and public health was overwhelmingly positive, especially compared to the unhygienic conditions prevalent elsewhere in the ancient world. Roman urban planning itself also contributed to public health. Well-designed streets, public spaces, and even regulations concerning building heights and ventilation, albeit rudimentary, aimed to create a more organized and healthier living environment. The state also took responsibility for ensuring a stable food supply, managing grain distribution to prevent famines, which were often a prelude to widespread disease. Another hallmark of Roman practicality in medicine was the development of organized military medicine. As an empire built on military might, ensuring the health and rapid recovery of its soldiers was paramount. The Romans established the valetudinaria, which were dedicated military hospitals, arguably the first true hospitals in history. These were not mere sick bays. They were well-structured facilities, often built adjacent to legionary forts, providing designated areas for surgery, convalescence, and even dedicated medical personnel. Each legion had its own medical corps, consisting of surgeons medici or chirurgi, physicians, and orderlies. They were trained in battlefield medicine, performing amputations, wound care, and managing injuries with surprising effectiveness. Roman surgical instruments, discovered through archaeological finds, demonstrate a high level of craftsmanship and variety, indicating sophisticated procedures. Furthermore, military encampments were meticulously planned with hygiene in mind, often selecting elevated ground, ensuring access to clean water, and managing waste, all to minimize disease among the troops. The vast experience gained from treating thousands of battlefield casualties led to significant advancements in trauma care and surgical techniques that would influence medical practice for centuries. While Roman contributions to public health and military medicine were profoundly practical, their theoretical medical knowledge largely derived from and was dominated by the Greek tradition. The most influential figure of this era, and indeed for over 1,000 years following, was Claudius Galen, 129 c. 216 CE. A Greek physician born in Pergamon, Galen studied extensively in Alexandria, the intellectual hub of the time, and later became the personal physician to several Roman emperors, including Marcus Aurelius. Galen was a prolific writer, a brilliant synthesist of existing medical knowledge, and a dedicated experimenter, particularly through animal dissection, as human dissection was largely forbidden. He meticulously described the anatomy of various animals, extrapolating his findings to humans, which, while often accurate, sometimes led to errors, e.g., describing the human liver as having 5 lobes like a pig's. He expanded upon the Hippocratic humoral theory, integrating it with Aristotelian philosophy, and his own detailed anatomical and physiological observations to create a comprehensive, highly detailed medical system. His work, including on the usefulness of the parts of the body and method of medicine, codified virtually all medical knowledge of his time. Galen emphasized rational diagnosis based on pulse, urine, and observation of symptoms, and prescribed a wide array of remedies, including herbs, minerals, and complex polypharmaceutical concoctions. He believed in the importance of lifestyle, diet, and exercise. His intellectual dominance was so profound that his theories became dogma for the next 1,300 years. His encyclopedic works were meticulously copied and preserved, becoming the authoritative texts for medical education throughout the Byzantine Empire, the Islamic Golden Age, and medieval Europe. While his comprehensive system provided a much-needed framework and preserved much ancient knowledge, his authoritative status also inadvertently stifled independent inquiry and anatomical dissection for centuries, as questioning Galen was akin to questioning truth itself. In summary, Roman medicine, while building upon Greek philosophical foundations, carved its own distinct path defined by a pragmatic, engineering-focused approach. Their unparalleled achievements in public health infrastructure, aqueducts, sewers, baths transformed urban living, and significantly reduced the burden of disease. Their organized military medicine established the world's first true hospitals and advanced trauma care. And through the monumental work of Galen, they synthesized, expanded, and preserved the vast body of ancient medical knowledge, which, despite its eventual scientific inaccuracies, provided the fundamental medical curriculum for the entire Western world and beyond for more than a millennium. Rome's legacy reminds us that medicine is not solely about treating individual illness, but also about creating a healthy environment that supports the well-being of entire populations. The fall of the Western Roman Empire in 476 CE marked a profound shift in European civilization, initiating a period often characterized as the Dark Ages in the West. As urban centers declined, intellectual life fragmented, and the structured medical systems of Rome largely dissipated. This era in Europe, spanning roughly from the 5th to the 15th centuries, saw medical knowledge become largely preserved within monastic orders and folk traditions, while simultaneously a vibrant and dynamic intellectual movement in the Islamic world was not only preserving, but significantly advancing medical science. This period, therefore, represents a fascinating dichotomy, a slowing of medical progress in Europe, juxtaposed with an explosion of innovation and scholarship in the East. In medieval Europe, medicine primarily retreated into the monasteries. Monks, acting as scribes, meticulously copied classical texts, including those of Hippocrates and Galen, albeit often selectively and with interpolations. Their focus was often on spiritual well-being alongside physical care, viewing illness as a test from God or a consequence of sin. Monastic infirmaries provided basic care for the sick and poor, reflecting Christian principles of charity. Herbal medicine, often documented in herbals, became the staple of treatment, drawing from local flora and inherited knowledge. However, systematic scientific inquiry, anatomical dissection, and experimental observation, which characterized the classical era, largely ceased. Medical theory remained heavily reliant on the humoral theory, as interpreted through the lens of Galen, and treatments were often limited to prayer, bloodletting sometimes excessively, herbal remedies, and purges. Surgery in Europe during this period often fell into the hands of barber-surgeons craftsmen who performed procedures like tooth extractions, wound dressing, and amputations, often without formal medical training and largely disconnected from the scholarly physicians. Superstition, astrology, and a blend of Christian doctrine permeated medical practice, making the understanding and treatment of disease a complex mix of the practical, the spiritual, and the often misinformed. The devastating arrival of the Black Death in the mid-14th century, which wiped out an estimated one-third to half of Europe's population, brutally exposed the limitations of contemporary medical understanding and practice. Physicians, lacking knowledge of contagion or pathology, were largely powerless against the plague, resorting to ineffective treatments and attributing the catastrophe to divine wrath or astrological alignments. Despite these limitations, seeds of future growth were sown in the later medieval period. The establishment of early universities, such as Salerno in Italy, often considered the first medical school in Europe, and later Bologna, Paris, and Montpellier began to formalize medical education. These institutions, however, still relied heavily on scholastic interpretations of Galen and Avicenna, whose work would become known through translation, emphasizing theoretical knowledge over practical experience or independent research. Dissections were rare, often limited to public demonstrations of animal anatomy or human anatomy based on Galenic texts, rather than discovery. In stark contrast to the European landscape, the Islamic world during its Golden Age, roughly from the 8th to the 13th centuries, experienced an unparalleled efflorescence of scientific and medical knowledge. Driven by a thirst for knowledge, a commitment to scholarship, and the need to care for a vast and diverse empire, Islamic scholars embarked on a monumental translation movement, systematically gathering and rendering into Arabic the works of Greek, Roman, Persian, and Indian scholars. Crucially, they did not merely preserve these texts, they critically engaged with them, expanded upon them, and built new knowledge upon their foundations. The achievements of Islamic medicine were profound and multifaceted. One of the most significant contributions was the development of the Bimaristan, or public hospital. far beyond the charitable infirmaries of Europe or the military valetudinaria of Rome. These institutions were sophisticated centers of healing, medical education, and research. Bimaristans were often grand complexes with separate wards for different illnesses, e.g., surgical, medical, ophthalmic, mental health, teaching facilities, pharmacies, libraries, and even lecture halls. They employed male and female physicians, nurses, and administrators, providing care for all regardless of social status or religion. This model of the specialized, public hospital was a revolutionary concept that would eventually influence the development of hospitals in Europe. Islamic pharmacology flourished, becoming arguably the most advanced of its time. Scholars meticulously cataloged and experimented with medicinal plants, minerals, and animal products from across their vast empire, introducing new drugs like senna, camphor, and musk. They developed sophisticated methods for drug preparation, including distillation and sublimation, and established professional pharmacies with regulatory standards. Figures like Ibn al-Badr documented hundreds of drugs, often with their botanical and medicinal properties. This systematic approach to compounding and dispensing laid the groundwork for modern pharmacy. The realm of surgery also saw remarkable advancements. Abulqasis Abu al-Qasim al-Zahrawi, a 10th-century Andalusian physician, authored Al-Tasrif, a 30-volume medical encyclopedia that included a detailed section on surgery. This text described over 200 surgical instruments, many of his own invention, and provided meticulous instructions for procedures such as cauterization, lithotomy removing bladder stones, surgical treatment of fractures, and even early forms of plastic surgery. He emphasized the importance of anatomy and dissection for surgeons, and is credited with introducing the use of ligatures for blood vessels, reducing blood loss. Prominent figures illuminated this golden age. Muhammad ibn Zakaria al-Razi, known in the West as Rhazes c. 865 to 925 CE, was a Persian polymath and physician. He was a pioneer in clinical observation, advocating for experimental medicine and rejecting blind adherence to authority. His most famous work, Kitab al-Hawi, the Comprehensive Book of Medicine, was a vast medical encyclopedia. But he is particularly renowned for his clear distinction between smallpox and measles, a crucial diagnostic breakthrough, and his emphasis on the importance of the patient's psychological state. Perhaps the most influential figure was Ibn Sina c. 980 to 1037 CE, known as Avicenna in the West. His monumental work, Al-Canon fi al-Tib, the Canon of Medicine, became the standard medical textbook in both the Islamic world and Europe for over 600 years. The canon synthesized Greek, Roman, and Indian medical knowledge with Islamic advancements, covering anatomy, physiology, diagnosis, pharmacology, and treatment. It described contagiousness of disease, introduced clinical pharmacology, and recognized the importance of diet and exercise. Avicenna also contributed significantly to the fields of anatomy, ophthalmology, and mental health. Islamic ophthalmology, in particular, saw significant progress, with detailed descriptions of eye diseases and sophisticated surgical techniques, including the earliest known cataract operations. They also made advances in understanding the digestive system, the nervous system, and the principles of hygiene. The transmission of this rich body of knowledge from the Islamic world back to Europe, often through centers like Toledo and Sicily, was gradual but transformative. Latin translations of Arabic medical texts, particularly Avicenna's canon and the surgical writings of Abulcasis, profoundly influenced the burgeoning medical schools in Europe from the 12th century onwards, breathing new life into a stagnant tradition and setting the stage for the Renaissance. The Dark Ages in Europe were thus illuminated by the radiant light of the Islamic Golden Age, which not only preserved ancient wisdom, but actively expanded medicine into a more sophisticated, systematic, and compassionate science, creating a vital bridge to modernity. The intellectual ferment of the Renaissance, a period spanning roughly from the 14th to the 17th centuries, is widely celebrated for its explosion in art, literature, and philosophy. Yet, beneath the surface of artistic genius and philosophical rediscovery, a profound transformation was also unfolding in the realm of medicine. This era marked a decisive turning point, a rebirth not merely of ancient texts, but of a critical, empirical spirit that would fundamentally reshape how physicians understood the human body and treated disease. For over a millennium, medical thought in the West had been largely dominated by the monolithic authority of Galen. His comprehensive anatomical and physiological theories, brilliant for their time, had become unchallengeable dogma, often perpetuated by rote learning from texts rather than direct observation. The Renaissance, however, brought a renewed emphasis on direct experience, observation, and a willingness to question even the most revered authorities, setting the stage for the true dawn of scientific medicine. At the epicenter of this anatomical revolution stands Andreas Vesalius, 1514 to 1564, a Flemish physician whose work irrevocably shattered the Galenic paradigm. Born in Brussels, Vesalius studied medicine in Paris and Louvain before becoming a professor of surgery and anatomy at the prestigious University of Padua, a center known for its progressive intellectual environment. It was here that Vesalius embarked on a radical departure from traditional medical teaching. For centuries, anatomy lectures typically involved a professor reading from Galen's texts, while a barber surgeon performed a crude dissection, often on an animal, with little actual engagement from the physician-lecturer. Vesalius, however, insisted on performing the dissections himself, directly demonstrating to his students the intricate structures of the human body. He believed firmly that true knowledge could only be gained through direct observation and manipulation, not through blind adherence to ancient texts. This hands-on approach quickly revealed discrepancies between Galen's descriptions and the actual human anatomy. Galen, having been largely restricted to animal dissections, primarily pigs and monkeys, due to Roman prohibitions on human dissection, had extrapolated many features to humans, leading to numerous errors. For instance, Galen believed the human sternum consisted of 7 parts, like a monkey's, when it has 3. He described a 5-lobed liver in humans, like a pig's, instead of the 2-lobed human liver. He also perpetuated the existence of the reedy mirabile, a complex network of blood vessels found at the base of the brain in some animals, but not in humans. These were not minor inaccuracies. They were fundamental misrepresentations of human structure. Vesalius' monumental achievement culminated in the publication of his masterpiece in 1543, De Humani Corporis Fabrica Libri Septem, on the fabric of the human body in 7 books, commonly known as the Fabrica. This was more than just an anatomical textbook. It was an artistic and scientific triumph. The Fabrica contained meticulously detailed and exquisitely rendered woodcut illustrations, likely produced by Jan van Kalkar, a student of Titian. These anatomical plates were revolutionary, depicting the human body with unprecedented accuracy, dynamism, and artistic flair. They showed muscles in action, bones articulated with lifelike precision, and organs intricately detailed. The combination of detailed, accurate text derived from direct observation and stunning visuals made the Fabrica an instant sensation and the most accurate anatomical atlas for centuries. The impact of the Fabrica was profound. It not only corrected hundreds of Galen's errors, but, more importantly, instilled a new methodological standard. That anatomical knowledge must be derived from direct human dissection and observation, not from ancient authority. It marked the definitive shift from textual authority to empirical evidence, a foundational principle of the scientific revolution that was concurrently unfolding. Vesalius's work was a bold declaration that understanding the human body required seeing and experiencing it directly. While Vesalius revolutionized anatomy, other figures pushed the boundaries of medical thought in different directions, driven by the same spirit of questioning and observation. Philippus Aureolus Theophrastus Bombastus von Hohenheim, better known as Paracelsus, 1493 to 1541, was a highly controversial and often abrasive Swiss-German physician and alchemist. He vehemently rejected the traditional Galenic humoral theory and the emphasis on scholastic learning. Instead, Paracelsus advocated for a medicine rooted in observation, experience, and what he termed iatrochemistry, the application of chemistry to medicine. Paracelsus believed that diseases were not merely imbalances of humors, but localized entities caused by specific external agents. Therefore, the cure should also be specific, a chemical remedy directed at the cause of the disease. He championed the use of minerals and chemical compounds in medicine, such as mercury for syphilis and sulfur for skin ailments, moving away from the traditional herbal remedies. Although many of his methods were crude, and his theories often mystical, his insistence on direct observation, his challenge to ancient authority, and his pioneering use of chemical substances in therapy marked a significant departure from established practices and laid an important groundwork for the future development of pharmacology. Building upon the foundations laid by Vesalius's anatomical precision and the Renaissance's empirical spirit, the English physician William Harvey, 1578 to 1657, made perhaps the most significant physiological discovery of this period, albeit published slightly after the strict chronological boundaries of the Renaissance. Harvey, who also studied at the University of Padua, applied meticulous observation, experimentation, and quantitative reasoning to understand the cardiovascular system. For over a millennium, Galen's theory of blood movement had held sway. That blood was consumed by the body and continuously produced by the liver, with blood flowing from one side of the heart to the other through invisible pores in the septum. Harvey challenged this. Through careful vivisections on animals, ligatures, and mathematical calculations, he demonstrated conclusively that blood circulated throughout the body in a closed system, pumped by the heart. In his groundbreaking work, Exercitatio Anatomica de Motu Cordis et Sanguinis in Animalibus, Anatomical Exercise on the Motion of the Heart and Blood in Animals, published in 1628, Harvey described the heart as a pump, propelling blood out through the arteries and receiving it back through the veins, thus establishing the concept of systemic circulation. His work was a triumph of empirical methodology, combining anatomical observation with physiological experimentation and quantitative reasoning, fundamentally transforming understanding of the body's vital processes. The rapid dissemination of these revolutionary ideas was also significantly aided by a key Renaissance innovation: the printing press. Invented by Johannes Gutenberg in the mid-15th century, the printing press made it possible to produce books, including medical texts and anatomical atlases like the Fabrica, in large quantities, and with unprecedented accuracy of illustration. This meant that Vesalius' detailed anatomical drawings and Harvey's physiological findings could reach a much wider audience of scholars, students, and practitioners across Europe, accelerating the pace of medical discovery and education. Knowledge, once confined to a few monastic libraries or painstaking handwritten copies, could now be widely distributed, fostering a more dynamic and interactive intellectual environment. In conclusion, the Renaissance was a crucible for medical progress. It was a period defined by a courageous willingness to look, to question, and to challenge centuries of entrenched dogma. Andreas Vesalius fundamentally transformed anatomy by insisting on direct human dissection and meticulous illustration, replacing ancient errors with empirical truth. Paracelsus, in his own controversial way, began to shift medicine towards a chemical understanding of disease and treatment. And William Harvey, exemplifying the peak of Renaissance empirical inquiry, laid bare the mysteries of blood circulation through rigorous experimentation. These pioneers, armed with a new spirit of inquiry and aided by the revolutionary printing press, did not just rediscover ancient knowledge. They actively interrogated the natural world, setting medicine on an irreversible course towards becoming an evidence-based, scientific discipline. This rebirth was not a mere looking back, but a forward leap, forever altering the trajectory of medical understanding. As the Renaissance propelled medicine forward with its emphasis on direct observation and human anatomy, the broader intellectual current of the scientific revolution, roughly spanning the 16th to 18th centuries, provided an even more profound shift in how humanity understood the natural world, including the intricacies of the human body. This period was characterized by a fervent commitment to empiricism, mathematical reasoning, and the development of new instruments that extended the very limits of human perception. In medicine, This era ushered in two transformative areas: the astonishing unveiling of the microscopic world and the nascent efforts to understand the mechanisms of bodily function, moving beyond mere anatomical description to a more dynamic understanding of physiology. For millennia, the universe of the incredibly small remained utterly hidden. Disease was attributed to imbalances of humors, miasmas, or divine intervention. No one could conceive of life too tiny to be seen. This blind spot was dramatically illuminated by the invention and refinement of the microscope. While the precise invention is debated, often attributed to Dutch spectacle makers like Hans and Zacharias Janssen around 1590, or even Galileo in 1609, adapting his telescope, its impact on biology and medicine was nothing short of revolutionary. One of the most seminal figures in revealing the microscopic realm was Robert Hooke, 1635 to 1703, an English natural philosopher and polymath. In 1665, Hooke published his monumental work, Micrographia, a book filled with stunningly detailed illustrations of microscopic observations. Using a compound microscope of his own design, he meticulously depicted the intricate structure of a flea, the stinger of a bee, and most famously, slices of cork. It was in observing cork that Hooke noticed tiny box-like structures, which he termed cells, because they reminded him of the small rooms, or cells, in a monastery. While Hooke saw only the dead cell walls, his observation was the first glimpse into the fundamental building blocks of life, laying the groundwork for cell theory centuries later. His work demonstrated the potential of the microscope to reveal previously unimaginable biological complexity. However, it was the Dutch draper, Antony van Leeuwenhoek, 1632 to 1723, who truly brought the microscopic world to life with an astonishing level of detail. Unlike Hooke, Leeuwenhoek was not a professional scientist. He was an amateur with an obsessive passion for grinding his own single-lens microscopes. And what lenses they were! His simple, yet incredibly powerful, microscopes, capable of magnifying objects up to 270 times, far surpassed the capabilities of compound microscopes of his time. With these tiny lenses, Leeuwenhoek became the first human to witness a menagerie of previously invisible life. From 1674 onwards, Leeuwenhoek began sending enthusiastic letters to the Royal Society of London, detailing his incredible discoveries. He was the first to observe and describe bacteria, which he called animalcules, found in pond water, tooth scrapings, and even fecal matter. He meticulously described the movements and shapes of these tiny creatures, providing the first concrete evidence of a living world beyond human sight. He also made the first accurate observations of red blood cells, describing their biconcave disc shape, and correctly identified sperm cells, even offering an early, albeit incorrect, theory of fertilization. Crucially, in 1683, Leeuwenhoek was the first to observe capillaries, the tiny blood vessels, connecting arteries and veins, confirming William Harvey's theory of blood circulation by providing the missing link. Leeuwenhoek's discoveries were not immediately integrated into mainstream medical theory, primarily because the sheer scale of these animalcules, made it difficult for people to grasp their significance in disease. However, his work irrevocably changed humanity's perception of life itself. It introduced the concept of a hidden, vibrant world teeming with microscopic organisms, a foundational revelation that would, much later, provide the critical insight for the germ theory of disease. The microscope, in the hands of Hooke, and especially Leeuwenhoek, shattered the belief that the visible world was all that existed, opening up an entirely new frontier for biological and medical investigation. Concurrent with the unveiling of the microscopic, the scientific revolution fostered a new approach to understanding the function of the human body, moving beyond descriptive anatomy towards dynamic physiology. Building on William Harvey's discovery of blood circulation, which established a mechanical model for a vital bodily process, thinkers began to view the body less as a mysterious, divinely animated entity, and more as a complex machine governed by physical and chemical laws. René Descartes' 1596 to 1650, the French philosopher, while not a physician, profoundly influenced this mechanistic view of the body. In his work Discourse on Method, he posited that the human body, unlike the mind-soul, was a machine, a complex automaton operating according to the laws of physics. While his dualism separated mind and body, his mechanistic view of the body encouraged a reductionist approach, suggesting that bodily functions could be understood by breaking them down into their component parts and analyzing their mechanical interactions. This philosophical perspective found its medical champion in Giovanni Alfonso Borelli, 1608 to 1679, an Italian physiologist, physicist, and mathematician. Borelli is considered the father of biomechanics for his pioneering work in applying the principles of mechanics to biological movement. In his posthumously published masterpiece, De Motu Animalium, on the movement of animals in 1680 to 81, he meticulously analyzed the musculoskeletal system. He calculated the forces involved in muscle contraction, viewing muscles as levers and bones as fulcrums. He demonstrated the mechanics of flying in birds, swimming in fish, and walking and running in humans, using mathematical precision. Borelli's work was a seminal moment in physiology, as it demonstrated that bodily functions could be explained by the same physical laws that governed inanimate objects, paving the way for quantitative and experimental physiology. Other early physiologists began to investigate various bodily functions with this new mechanistic lens. Richard Lower, 1631 to 1691, an English physician, further elucidated the process of respiration, demonstrating that blood changed color in the lungs due to exposure to air, an early step towards understanding oxygenation. Jean Pecquet, 1622 to 1674, a French anatomist, correctly identified the thoracic duct and its role in the lymphatic system, revising earlier understandings of how digested nutrients entered the bloodstream. This era saw a shift from simply describing what the body parts were to investigating how they worked. While many of the early physiological theories were still rudimentary and often speculative, the critical methodology was established. To apply observation, experimentation, and, where possible, mathematical reasoning to unravel the mysteries of living systems. The humoral theory, though still dominant in clinical practice, was increasingly challenged by new anatomical and physiological insights that offered more concrete, physical explanations for bodily processes. In essence, the scientific revolution provided medicine with two indispensable lenses. The microscope allowed humanity to peer into an invisible world, revealing organisms that would eventually be understood as agents of disease, and demonstrating the intricate cellular architecture of life. Simultaneously, the application of mechanical principles to the human body by early physiologists began to unravel the complex functions of organs and systems, transforming the body from a mystical entity into a subject of scientific inquiry, a sophisticated machine amenable to logical investigation. These dual advancements laid crucial methodological and conceptual foundations, setting the stage for the dramatic medical breakthroughs of subsequent centuries, particularly in microbiology and modern biological research. The Age of Enlightenment, a transformative intellectual and philosophical movement of the 18th century, fundamentally reshaped the Western world's understanding of reason, nature, and society. Driven by a fervent belief in logic, empirical evidence, and the power of human intellect to solve problems, this era extended its profound influence into the nascent science of medicine. Moving beyond the anatomical revelations of the Renaissance and the early physiological insights of the scientific revolution, the Enlightenment ushered in a critical emphasis on systematic clinical observation and the classification of diseases. This period was characterized by a concerted effort to organize medical knowledge, to meticulously document what was seen at the patient's bedside, and crucially, to correlate those clinical observations with the underlying changes within the body, marking a vital step towards a more scientific and less speculative approach to healing. For centuries, medical diagnosis had been a largely intuitive art, heavily influenced by the abstract humoral theory or the often dogmatic interpretations of ancient texts. Treatments were often generalized, aimed at restoring a perceived systemic balance rather than targeting specific ailments. The Enlightenment, however, demanded a more precise and methodical approach. Physicians began to realize that true understanding of disease required not just knowing the parts of the body anatomy or how they generally functioned physiology, but how disease specifically manifested in living patients and how it altered the body's internal structure. This intellectual shift gravitated towards the bedside and the autopsy table, linking the visible symptoms to the invisible internal pathology. One of the most influential figures embodying this new spirit of clinical observation was Thomas Sydenham, 1624 to 1689, often called the English Hippocrates, for his unwavering commitment to direct patient observation. Sydenham lived slightly before the peak of the Enlightenment, but his principles profoundly influenced 18th century medical thought. He rebelled against the prevalent theoretical speculation and rigid adherence to ancient texts. Instead, he argued that physicians should meticulously observe and record the natural history of diseases, much like a botanist classifies plants. He believed that diseases were distinct species, each with its own characteristic symptoms, course, and response to treatment. Sydenham meticulously documented symptoms, noting their onset, progression, and resolution. He observed patterns in epidemics, correlating environmental factors and seasonal variations with the prevalence of certain diseases. For example, he provided the classic descriptions of diseases like measles, scarlet fever, and gout, differentiating them by their distinct clinical presentations. He emphasized that the physician's role was to support the body's natural healing processes, advocating for simple remedies and a less interventionist approach than was common at the time, often critical of excessive bloodletting or complex polypharmacy. His work fundamentally shifted the focus from abstract internal imbalances to the tangible, observable manifestations of disease, laying the groundwork for clinical epidemiology and the concept of disease entities. His unwavering empiricism and dedication to the patient's lived experience made him a guiding light for future clinicians. The systematic approach to understanding disease took another monumental leap forward with Giovanni Battista Morgagni, 1682 to 1771, an Italian anatomist and pathologist. While Sydenham focused on the clinical picture of disease, Morgagni bridged the gap between symptoms observed in the living and the physical changes found in the deceased. His magnum opus, published in 1761, was titled De Sedibus et Causes Morborum per Anatomen Indagatis, which translates to On the Seats and Causes of Diseases Investigated by Anatomy. This book was a landmark in medical history, fundamentally establishing the field of pathological anatomy. Morgagni meticulously correlated clinical histories taken from his patients with his findings from hundreds of post-mortem dissections. He demonstrated that diseases were not merely generalized imbalances affecting the whole body, but were often localized to specific organs or tissues. For instance, he might describe a patient's symptoms of breathlessness and coughing clinical observation, and then, upon autopsy, reveal a specific lesion in the lungs that explained those symptoms' pathological anatomy. This was a revolutionary concept that began to dismantle the holistic humoral theory piece by piece. His work provided concrete, anatomical explanations for a wide range of ailments, from heart disease and aneurysms to liver disorders and brain lesions. Morgagni's methodology transformed the autopsy from a mere anatomical demonstration into a powerful diagnostic and educational tool. It solidified the idea that diseases had a physical basis, a seat within the body, which could be investigated and understood. This established a new paradigm for medical inquiry. Observe the patient, record the symptoms, hypothesize about the internal cause, and then, if the patient died, confirm or refute that hypothesis through post-mortem examination. This rigorous correlation between clinical symptoms and anatomical findings laid the direct foundation for modern pathology and significantly advanced the understanding of disease processes. Inspired by the success of classification systems in natural history, particularly Carl Linnaeus's systematic taxonomy of plants and animals, Enlightenment thinkers also sought to apply similar principles to diseases. This led to the development of nosology, the systematic classification and naming of diseases. The goal was to bring order to the bewildering array of human afflictions, to create a common language for diagnosis, and to facilitate the collection and analysis of medical data. Early nosologists, such as François Boissier de Sauvages de Lacroix, who published Nosologia Methodica in 1768, attempted to categorize diseases into classes, orders, genera, and species, much like botanical classifications. While these early systems were imperfect, often based on symptoms rather than underlying causes which were still largely unknown, and sometimes created arbitrary distinctions, they represented a crucial step towards standardizing medical terminology and creating a framework for medical knowledge. The idea was that if diseases could be precisely identified and categorized, then more effective and specific treatments could be developed, and patterns of disease occurrence, early epidemiology, could be better understood. This urge to classify, to bring rational order to the apparent chaos of illness, reflected the Enlightenment's broader scientific ambitions. These methodological shifts were increasingly integrated into medical education, particularly in the emerging hospital-based clinical schools, most notably in Vienna and Edinburgh, and later the renowned Paris Clinical School of the late 18th and early 19th centuries. Here, students were no longer confined to lecture halls, but were actively engaged at the patient's bedside, taught to observe, examine, and record. The hospital became a living laboratory where clinical signs were linked to pathological findings, solidifying the principles established by Sydenham and Morgagni. This practical, hands-on training, combining clinical skill with anatomical knowledge, was a monumental advance over the purely theoretical instruction of earlier centuries. Despite these profound methodological advancements, it is important to note that actual treatments during the Enlightenment were still largely limited. Physicians possessed a much better understanding of what a disease looked like and where it resided, but the fundamental causes like germs or specific cellular dysfunctions remained largely unknown. Therefore, treatments often continued to be empirical and sometimes harmful, including bloodletting, purges, and the administration of various elixirs. However, the approach had fundamentally changed. The move from abstract speculation to concrete, systematic observation, and the meticulous correlation of symptoms with anatomical lesions created an indispensable intellectual framework. In conclusion, the Age of Enlightenment, through its emphasis on reason, empirical evidence, and classification, profoundly reshaped medicine. Thomas Sydenham's unwavering commitment to detailed clinical observation and the concept of distinct disease entities provided a new way to understand illness. Giovanni Battista Morgagni, with his pioneering work in pathological anatomy, established the vital link between clinical symptoms and internal organic changes, effectively making the autopsy a crucial diagnostic tool. The burgeoning field of nosology, though imperfect, reflected the era's drive to bring systematic order to medical knowledge. Together, these developments laid the crucial intellectual and methodological groundwork, shifting medicine irrevocably towards an evidence-based discipline, setting the stage for the dramatic scientific breakthroughs of the 19th century that would finally unveil the hidden causes of disease. For millennia, humanity lived under the constant shadow of pestilence. Epidemics of infectious diseases swept through populations with terrifying regularity, decimating communities and shaping the course of history. Among the most feared and devastating of these scourges was smallpox. Characterized by a distinctive rash that left survivors scarred or blind, smallpox was a relentless killer, claiming millions of lives across continents for thousands of years. Its mortality rate was staggering, often exceeding 30 in general populations and far higher in infants. Those who survived often bore lifelong disfigurement, a grim testament to the disease's ferocity. The desperate need for a way to prevent, rather than merely treat, such a pervasive and lethal threat spurred one of medicine's most profound breakthroughs. The development of preventive medicine, specifically the journey from rudimentary inoculation to sophisticated vaccination. The earliest glimmer of an effective preventive strategy against smallpox emerged not from the scientific laboratories of Europe, but from ancient folk practices in Asia. This technique, known as variolation or inoculation, involved deliberately exposing a healthy person to material from a mild case of smallpox, in the hope of inducing a less severe form of the disease and subsequent immunity. Evidence suggests forms of variolation were practiced in China as early as the 10th century and in India by the 17th century. The Chinese method, for instance, often involved blowing dried smallpox scabs up the nostril, while in other regions, a small amount of pus from a fresh smallpox lesion might be scratched into the skin. The underlying principle, though not understood in germ theory terms, was a rudimentary form of immunotherapy. By introducing a small, controlled amount of the pathogen, the body would mount a response and, if successful, develop immunity to future, more severe exposures. While this was a radical and often risky proposition, the results, in many cases, were better than natural infection. Variolation typically resulted in a milder case of smallpox, with a mortality rate of around 1 to 2, significantly lower than the 20 to 30 or higher death rate from naturally acquired smallpox. The knowledge of variolation slowly filtered westward. A pivotal figure in its introduction to Europe was Lady Mary Wortley Montagu. As the wife of the British ambassador to the Ottoman Empire, she observed the practice of variolation in Constantinople in 1717. Deeply impressed by its efficacy, especially after seeing its benefits firsthand and having her own son variolated, she became a fervent advocate. Upon her return to England, she championed the practice, notably having her daughter variolated in 1721 by Charles Maitland, a physician who had accompanied her to Turkey. This act, audacious for its time, brought variolation to the attention of the British elite, and the royal family, slowly gaining acceptance, despite considerable initial skepticism and opposition, often from religious authorities who viewed it as interfering with divine will. While variolation offered a significant reduction in mortality compared to natural smallpox, it was far from perfect. It still involved deliberately giving a person smallpox, meaning there was a risk of severe disease or even death. Furthermore, variolated individuals, though often having a milder illness, were infectious and could transmit the full-blown disease to others, potentially sparking new outbreaks. This inherent risk limited its widespread adoption and made it a public health concern in itself. A safer, more universally applicable method was desperately needed. This is where the pioneering work of Edward Jenner, 1749 to 1823, an English country doctor, comes into play, forever changing the course of preventive medicine. Jenner's breakthrough was a testament to meticulous observation, intellectual curiosity, and rigorous experimentation qualities that would come to define modern scientific inquiry. For years, Jenner had observed a common folk belief in rural Gloucestershire. Milkmaids who contracted cowpox, a mild disease of cattle transmitted to humans through contact with infected udders, seemed to be immune to smallpox. They might get a few lesions on their hands, but they never succumbed to the dreaded smallpox. This anecdotal evidence sparked a profound hypothesis in Jenner's mind. Could cowpox provide protection against smallpox? Jenner decided to test this hypothesis empirically. On May 14, 1796, he performed his seminal experiment. He took pus from a cowpox lesion on the hand of a milkmaid named Sarah Nelmes and inoculated an 8-year-old boy named James Phipps by scratching the material into his arm. As expected, James developed a mild fever and a few localized lesions, but quickly recovered. Several weeks later, Jenner subjected James to a far more dangerous challenge. He inoculated the boy with material from a genuine smallpox lesion. To Jenner's immense relief and scientific triumph, James Phipps showed no signs of smallpox infection. He was protected. This experiment demonstrated a critical distinction from variolation. Protection could be achieved using a different, much milder virus cowpox, or variolae vaccinae, as it was then known, which did not cause severe disease in humans and did not lead to infectious smallpox. Jenner termed his new procedure vaccination, derived from the Latin word vacca for cow. Jenner published his findings in 1798 in a concise but revolutionary treatise titled An Inquiry into the Causes and Effects of the Variolae vaccinae, a disease discovered in some of the western counties of England, particularly Gloucestershire, and known by the name of the cow-pox. Initially, his work faced resistance and skepticism from the medical establishment. Variolation proponents viewed him with suspicion, and others found the idea of introducing animal disease into humans repulsive. Religious objections also arose, with some arguing it was unnatural or an affront to God. However, the compelling evidence of vaccination's safety and effectiveness gradually overcame these objections. Unlike variolation, vaccination did not carry the risk of death from the procedure itself, nor did it cause the spread of human smallpox. Its benefits were clear and undeniable. Over the next few decades, vaccination rapidly spread across Europe and then globally, championed by figures like Napoleon Bonaparte, who ordered his entire army vaccinated. Its adoption was a testament to its undeniable success in protecting against smallpox, leading to a dramatic decline in smallpox mortality and incidence wherever it was widely implemented. The impact of Jenner's vaccination was monumental. It marked the first truly effective and safe method of preventing a widespread infectious disease. It demonstrated that immunity could be artificially induced, a concept that laid the foundational stone for the entire field of immunology. Jenner's empirical success, achieved decades before Louis Pasteur would articulate the germ theory of disease, proved that diseases could be controlled on a population scale through public health interventions, not just individual treatments. It shifted the medical paradigm from reactive treatment to proactive prevention. The success of vaccination spurred future research into infectious diseases and the mechanisms of immunity. It inspired scientists like Pasteur to seek out other attenuated forms of pathogens for vaccine development. The ethical challenges of early human experimentation, evident in Jenner's testing on James Phipps, also laid the groundwork for future discussions on informed consent and patient safety in medical research. The global campaign for smallpox eradication, spearheaded by the World Health Organization in the 20th century, stands as the crowning achievement of Jenner's legacy. Through relentless vaccination campaigns, smallpox was declared officially eradicated in 1980, the only human disease to have been completely wiped out from the face of the earth. This triumph stands as a powerful testament to the vision of a country doctor who, through careful observation and courageous experimentation, transformed the fight against infectious diseases, setting humanity on the path towards a future where many once deadly scourges could be prevented. The journey from crude inoculation to systematic vaccination encapsulates the very essence of progressive medical thought. The constant quest for safer, more effective, and more widely applicable solutions to humanity's most pressing health challenges. For much of human history, the act of healing was a largely individual endeavor, conducted in homes, by local healers, or within the confines of private practices. The institutions we now recognize as hospitals, vast centers of medical care, teaching, and research, are a relatively modern phenomenon, having undergone a profound transformation from their humble origins. This evolution from places of charity and isolation to indispensable scientific hubs is intrinsically linked to the parallel professionalization and scientific grounding of medical education itself. Early forms of collective care emerged in various ancient civilizations, though they bore little resemblance to modern hospitals. In ancient Rome, valetudinaria served as dedicated military infirmaries, providing structured care for wounded soldiers. In the Byzantine Empire and later in medieval Europe, monastic infirmaries and hospices, often run by religious orders, offered shelter, food, and basic comfort to the sick, the poor, and travelers. These institutions, however, were primarily driven by spiritual and charitable impulses, viewing physical suffering as an opportunity for spiritual salvation. While some basic remedies might be administered, systematic medical treatment, anatomical study, or formal clinical education were rarely their primary focus. Patients often entered these places to die, or to recover through rest and prayer, rather than to receive advanced medical intervention. A crucial, though often overlooked, precursor to the modern hospital model emerged in the Islamic Golden Age. The Bimaristan, meaning House of the Sick in Persian, was a revolutionary concept. From the 9th century onwards, These institutions in cities like Baghdad, Cairo, and Damascus were far more than simple infirmaries. They were sophisticated, multi-purpose centers offering comprehensive medical care, segregated wards for different diseases e.g., surgical, internal medicine, mental illness, and employing salaried physicians, nurses, and pharmacists. Crucially, Bimaristans were also centers of medical education, serving as teaching hospitals where students learned at the patient's bedside and through the study of extensive medical libraries. They were open to all, regardless of social status or religion, reflecting a commitment to public health and scientific inquiry that profoundly influenced later European developments. Back in medieval Europe, the charitable hospital model persisted. The Hôtel Dieu in Paris, founded in 651 CE, exemplifies this type of institution. While providing crucial shelter and basic care, these early European hospitals were often overcrowded, unsanitary, and lacked a systematic approach to diagnosis or treatment. The prevailing medical education, meanwhile, was largely theoretical, centered in emerging universities like Salerno, Bologna, and Padua. Students would primarily learn from lectures, often based on Latin translations of Galen and Avicenna. Clinical experience was minimal, and anatomical dissection, though slowly gaining ground in the Renaissance, was usually a rare public spectacle rather than a regular part of student training. A significant disconnect remained between the academic physician, who adhered to theory, and the practical barber surgeon, who performed manual procedures. The true transformation of the hospital into a scientific and educational epicenter began to accelerate in the 18th century, a period shaped by the Enlightenment's emphasis on empirical observation and systematic reasoning. The limitations of treating patients based solely on abstract theories became increasingly apparent. Physicians began to demand access to patients and their cases for direct observation and study. This era saw a conceptual shift. The hospital was no longer just a place to isolate the sick, but a living laboratory for understanding disease. Pioneering figures and institutions drove this change. In Vienna, during the mid-18th century, physicians like Gerard van Swieten and Anton de Heen, under the patronage of Empress Maria Theresa, established a more structured system of clinical instruction at the Allgemeines Krankenhaus General Hospital. Here, medical students began to spend more time at the bedside, observing patients, taking detailed histories, and learning physical examination techniques. This marked a significant departure from purely theoretical instruction. Similarly, the University of Edinburgh, with its Royal Infirmary, became a leading center for clinical teaching, attracting students from across Europe and the American colonies. However, the most profound and influential transformation occurred with the rise of the Paris Clinical School in the late 18th and early 19th centuries. This school represented the zenith of the hospital medicine movement. Following the French Revolution, many old charitable and religious hospitals were secularized and centralized, creating large institutions with unprecedented patient populations. This offered a unique opportunity for systematic clinical research and teaching. Leading figures of the Paris Clinical School, such as Philippe Pinel, Jean-Nicolas Corvisart, and especially René Laennec, revolutionized medical practice and education. They championed the methodical collection of detailed patient histories, regular ward rounds where students learned directly from patients, and rigorous physical examination. Corvisart, physician to Napoleon, emphasized percussion, tapping on the body, to assess underlying structures and auscultation, listening to internal body sounds. His student, Laennec, took auscultation to a new level by inventing the stethoscope in 1816, a simple yet revolutionary device that allowed physicians to listen to heart and lung sounds with unprecedented clarity, providing crucial diagnostic information that was previously unattainable. The stethoscope became a symbol of modern bedside diagnosis and a powerful teaching tool. Crucially, the Paris Clinical School institutionalized the practice of pathological autopsy. For the first time, it became routine to conduct post-mortem examinations on deceased patients and meticulously correlate the findings of organ damage and disease lesions with the symptoms observed during the patient's life. This direct link between clinical symptoms and underlying anatomical pathology, building on the work of Morgagni, was a cornerstone of modern diagnostic medicine. Hospitals, with their large patient populations and autopsy facilities, became indispensable for this process. This systematic correlation allowed physicians to refine their understanding of specific disease entities, moving away from vague generalities towards precise pathological diagnoses. The Paris Clinical School also introduced numerical medicine or statistical analysis, particularly championed by Pierre Louis. By analyzing large numbers of cases, Louis sought to determine the effectiveness of various treatments, such as bloodletting, through empirical data, rather than tradition or anecdote. This early application of statistical reasoning laid the groundwork for evidence-based medicine. The impact of this transformation was immense. Medical education shifted decisively from the lecture hall to the hospital ward. Students gained invaluable hands-on experience, learning to observe, diagnose, and treat directly from patients under the guidance of experienced clinicians. The physician's role evolved from a remote theorist to an engaged clinician scientist. Hospitals became the primary sites for advanced medical training, attracting students and doctors from around the world, spreading the model of clinical instruction. This evolution of hospitals as centers of both patient care and medical education laid the indispensable groundwork for the scientific breakthroughs of the later 19th century. When the germ theory of disease emerged, the hospital was already the established environment for scientific investigation, allowing for the rapid integration of aseptic techniques, surgical advancements, and the development of specialized departments. From being largely places of last resort or charitable confinement, hospitals became dynamic institutions at the forefront of medical research, innovation, and the training of generations of medical professionals. The rise of the modern hospital and the integration of clinical education fundamentally professionalized medicine, establishing the rigorous standards of practice and learning that define healthcare today. For millennia, the very thought of surgery evoked not merely discomfort, but abject terror. Before the mid-19th century, Undergoing a surgical procedure was an experience of unimaginable agony, a brutal spectacle endured with stoicism, or more commonly, with screams that echoed through the operating theater. Pain was the surgeon's greatest adversary, and speed was his most prized virtue. Operations were performed with lightning quickness, not out of skill, but out of grim necessity to minimize the torment of the conscious patient. Limbs were amputated in mere minutes, teeth yanked without pause, and superficial tumors excised, while the patient was held down by strong assistants. Internal surgery, exploring the delicate organs of the abdomen or chest, was virtually impossible. The sheer shock, pain, and involuntary movements of the patient rendered such interventions unfeasible and almost certainly fatal. Surgical success was often measured less by the outcome of the procedure and more by the sheer survival of the patient through the ordeal itself. The psychological trauma of such experiences haunted both patients and practitioners, and the vast potential of surgical intervention remained locked behind the impenetrable wall of excruciating pain. Throughout history, humanity had sought solace from pain, both in general life and during surgical procedures. Ancient civilizations employed various crude analgesics and sedatives: alcohol to dull the senses, opium from the poppy plant to induce stupor, mandrake root, cannabis, and even certain local anesthetics derived from plants. In ancient Egypt and Rome, there are records of using pressure on nerves, or even inducing brief unconsciousness through carotid compression. During the medieval period and into the early modern era, these methods persisted, often augmented by methods of physical restraint straps, ropes, or the sheer brute force of multiple assistants. The late 18th and early 19th centuries even saw the brief, controversial use of mesmerism, or animal magnetism, a form of hypnotism, to induce a trance-like state during minor procedures. While some patients reported reduced sensation, these methods were inconsistent, unreliable, and offered no guarantee of true unconsciousness or complete pain relief for the duration required by major surgery. The fundamental challenge remained: how to reliably, safely, and reversibly render a patient insensible to pain, allowing the surgeon to work with precision and deliberation. The initial steps towards truly effective anesthesia came not from the medical establishment, but from the burgeoning field of chemistry and, ironically, from recreational experimentation. In the late 18th century, pioneering chemists were isolating and experimenting with various gases. Joseph Priestley discovered nitrous oxide in 1772, and by the end of the century, the brilliant young chemist Humphrey Davy began to systematically investigate its properties. In 1799, at the Pneumatic Institution in Bristol, Davy inhaled large quantities of what quickly became known as laughing gas, documenting its intoxicating and analgesic effects. He noted its ability to produce pleasurable sensations and, critically, a loss of physical pain. In his 1800 publication, Researches, Chemical and Philosophical, chiefly concerning nitrous oxide, or dephlogisticated nitrous air, and its respiration, Davy presciently wrote, "As nitrous oxide, in its extensive operation, appears capable of removing pain, It may probably be used with advantage during surgical operations in which no great effusion of blood takes place. Despite this astonishing foresight, Davy's suggestion was largely ignored by the medical community, who viewed nitrous oxide as little more than a novelty, a curiosity for parlor tricks and social frolics. For decades, it would remain a recreational diversion, its profound medical potential overlooked. A similar narrative unfolded with diethyl ether. Known for centuries by alchemists and used as a solvent, its intoxicating properties were also recognized early on. By the 1830s and 1840s, ether frolics, or ether parties, became popular social events, particularly among medical students and young professionals in America. Participants would inhale ether for its stimulating and euphoric effects, often leading to uninhibited behavior, but critically, also demonstrating its ability to induce a state of oblivion or insensibility to pain. Stories circulated of participants falling, cutting themselves, or otherwise injuring themselves during these frolics, without feeling pain, until the effects of the ether wore off. The anecdotal evidence was there, widespread and observable, yet the leap from recreational amusement to formal medical application proved elusive, waiting for the right confluence of desperation, observation, and daring. The true pioneers of surgical anesthesia were not university professors or established surgeons, but rather dentists and general practitioners, often working at the periphery of the conventional medical establishment. The first documented surgical use of ether, though not immediately publicized, came from a rural Georgia physician named Crawford Williamson, long 1815 to 1878. Having observed the pain-dulling effects of ether during frolics, Long courageously decided to apply it in a surgical setting. On March 30, 1842, he successfully used ether to remove a tumor from the neck of a patient named James Venable. He continued to use ether in several other operations over the next few years, recognizing its immense potential. However, Long, being a modest country doctor, did not publish his findings until 1849, long after the news of ether's success had already electrified the medical world. While he holds the distinction of being the first to use ether surgically, his lack of immediate publication meant his discovery did not spark the revolution. The stage was thus set for a public demonstration that would forever alter the course of medicine. The next significant figure was Horace Wells, 1815 to 1848, a dentist from Hartford, Connecticut. Inspired by a public demonstration of nitrous oxide in 1844, where a man under its influence injured his leg but felt no pain, Wells immediately recognized its potential for painless tooth extractions. He successfully used it himself for his own tooth extraction and performed several others. Convinced of his discovery, Wells sought to demonstrate his method to the skeptical medical establishment in Boston in January 1845. His demonstration at Massachusetts General Hospital was, tragically, a public failure. The patient cried out during the tooth extraction, leading to ridicule and dismissing nitrous oxide's anesthetic potential for years to come. Wells, despondent, abandoned his pursuit. This setback, however, only fueled the determination of William Thomas Green Morton, 1819 to 1868, a former partner of Wells and also a dentist. Having witnessed Wells' failure, Morton was determined to find a more reliable anesthetic. He experimented with various substances, including opium and ammonia, before turning his attention to ether on the advice of his former chemistry professor, Charles Jackson. Morton spent weeks refining the ether delivery method, experimenting on animals and even himself. Convinced he had found the answer, Morton secured an invitation from the eminent surgeon Dr. John Collins Warren, 1778 to 1856, to demonstrate his new method at Massachusetts General Hospital. The date was October 16, 1846. In the now famous Ether Dome operating amphitheater, before a hushed crowd of skeptical surgeons, medical students, and observers, Morton administered his ether vapor to a patient named Gilbert Abbott, who required a tumor removed from his neck. As the ether took effect, Abbott slipped into a state of unconsciousness. Dr. Warren then began the incision. The crowd watched, mesmerized, as Abbott remained still without a cry or a twitch. When the operation was complete, Dr. Warren turned to the astonished audience and uttered the immortal words, "Gentlemen, this is no humbug." The demonstration was an unqualified success, marking the public birth of surgical anesthesia. News of the ether discovery spread like wildfire across the globe, electrifying the medical community and inspiring a torrent of experimentation and adoption. The immediate aftermath of Morton's triumph, however, was not without controversy. Morton attempted to patent his discovery under the name Lethean, leading to bitter disputes over priority and credit with Wells, Jackson, and even Crawford Long, who belatedly published his earlier use. This ether controversy raged for years, involving lawsuits, accusations, and public debates, tarnishing the reputations of many involved. Despite the personal squabbles, the undeniable efficacy of ether meant that its use spread rapidly, regardless of who claimed credit or whether it was patented. The humanitarian benefits were simply too profound to be contained. Close on the heels of ether's success came the introduction of chloroform. In November 1847, just over a year after Morton's demonstration, James Young Simpson, 1811 to 1870, a Scottish obstetrician, pioneered the use of chloroform for pain relief, particularly in childbirth. Chloroform offered several advantages. It was faster-acting, non-flammable, and required a smaller dose than ether. However, it also carried a narrower margin of safety and a higher risk of fatal cardiac arrest. Despite its risks, chloroform gained widespread acceptance, especially after Queen Victoria famously used it for the birth of her 8th child, Prince Leopold, in 1853, administered by the renowned physician John Snow, who would later become famous for his work on cholera. The royal seal of approval helped to overcome lingering religious and moral objections to pain relief during childbirth. The advent of anesthesia initiated nothing short of a revolution in surgery. For the first time, surgeons were liberated from the tyranny of time. They could now operate with deliberation, precision, and a new level of humane care. This allowed for the performance of far more complex and intricate procedures that were previously unimaginable. deep abdominal surgeries, operations on internal organs, prolonged bone repairs, and more extensive tumor removals. The fear and shock associated with surgery dramatically decreased, saving countless lives and reducing immense suffering. The ability to render a patient unconscious and pain-free paved the way for subsequent surgical advancements. It created the necessity for a new medical specialty, anesthesiology, a field dedicated to ensuring patient safety, monitoring vital signs, and managing the delicate balance of consciousness and pain during surgery. The pain revolution was not merely a technological innovation, it was a profound humanitarian leap. It fundamentally altered the relationship between physician and patient, ushering in an era where patient comfort became a paramount concern. It expanded the boundaries of what was medically possible, allowing surgery to evolve from a last resort of desperation into a powerful and often life-saving intervention. The discoveries of ether and chloroform stand as monumental achievements, on par with the later understanding of germ theory, in reshaping the landscape of modern medicine and dramatically improving the human condition. For millennia, disease remained an enigma, its origins shrouded in mystery. Before the mid-19th century, the dominant medical explanation for widespread illness, particularly in crowded urban centers, was the miasma theory. This theory posited that diseases like cholera, plague, and fevers were caused by bad air, noxious fumes, vapors, or corrupt atmospheric effluvia arising from decaying organic matter. While the observation that foul-smelling places were often unhealthy was accurate, the causation was entirely mistaken. This pervasive belief, however, directly hampered effective public health measures, as efforts were focused on air purification rather than sanitation or understanding the true mechanisms of transmission. Yet, even as the miasma theory held sway, two extraordinary individuals, through meticulous observation and courageous deduction, began to sow the crucial seeds of what would eventually blossom into the revolutionary germ theory of disease. Their work, though performed without the aid of microscopes powerful enough to see bacteria, provided compelling, irrefutable evidence that invisible agents, transferred through specific means, were responsible for devastating epidemics. Our first pivotal story takes us to the bustling and often grim hospitals of mid-19th century Vienna, specifically to the Vienna General Hospital in the 1840s. Here, a Hungarian physician named Ignaz Semmelweis (1818 to 1865) confronted a terrifying and baffling problem: puerperal fever, or childbed fever. This horrific infection ravaged new mothers, causing high fevers, abdominal pain, and often agonizing death within days of childbirth. The mortality rates were appalling, sometimes as high as 20 to 30 in some hospital wards, a tragic reality for women who had just brought new life into the world. Semmelweis, working in the first obstetrical clinic, was deeply troubled by an inexplicable disparity he observed. His clinic, where medical students were trained, consistently had a much higher maternal mortality rate from puerperal fever than the second clinic, which was used for the training of midwives. This stark difference, despite both clinics treating similar populations and being located in the same hospital, baffled everyone. Physicians, rooted in the miasma theory, speculated about various causes: changes in climate, overcrowding, even the fear induced by a priest who passed through the first clinic on his way to administer last rites. Semmelweis systematically investigated and eliminated each of these hypotheses, finding no statistical correlation. The crucial aha moment came in 1847 with the tragic death of his friend and colleague, Jakob Kolechka, a professor of forensic medicine. Kolechka died from an illness that strikingly resembled puerperal fever after a medical student accidentally punctured his finger with a scalpel during a dissection of a corpse. Semmelweis, with astonishing insight, connected the dots. He hypothesized that cadaverous particles, invisible, decaying organic matter from the cadavers being dissected in the anatomy lab, were being transferred to the birthing mothers on the unwashed hands of the medical students, who routinely moved between the dissecting room and the delivery ward without cleaning their hands. The midwives, conversely, did not participate in autopsies. Armed with this radical hypothesis, Semmelweis immediately implemented a dramatic intervention. In May 1847, he mandated that all medical students and doctors thoroughly wash their hands with a chlorinated lime solution, calcium hypochlorite, before examining pregnant women. Chlorinated lime was chosen for its ability to neutralize the putrid smell of cadaverous matter, but Semmelweis intuitively understood it possessed disinfectant properties. The results were immediate and astonishing. Within months, the mortality rate in the first clinic plummeted from over 10 to under 2, eventually matching and even falling below that of the second clinic. Semmelweis had found a simple, effective solution that saved countless lives. However, his triumph was met with widespread skepticism, ridicule, and outright hostility from the medical establishment. He could not explain what these cadaverous particles were, only that they existed and were transmitted. Many prominent physicians were deeply offended by the suggestion that they, as gentlemen and healers, were responsible for inadvertently killing their patients due to their unclean hands. Semmelweis's attempts to spread his discovery were largely thwarted, and he eventually returned to Hungary, a broken man, dying tragically in an asylum from an infection remarkably similar to puerperal fever, ironic given his life's work. His contributions would only be fully recognized decades later, with the advent of germ theory. While Semmelweis tackled the problem of person-to-person transmission within hospitals, another brilliant mind was busy deciphering the mysteries of disease transmission on a broader, community scale. John Snow, 1813 to 1858, an English physician and pioneering anesthesiologist famous for administering chloroform to Queen Victoria, was deeply skeptical of the prevailing miasma theory as the sole explanation for the devastating cholera epidemics that periodically swept through London and other major cities. Cholera, a terrifying disease causing rapid and severe dehydration, often killed within hours, leaving a trail of panic and death. In the summer of 1854, a particularly virulent outbreak of cholera struck the Soho district of London. While many fled, Snow embarked on a meticulous, systematic investigation that would establish him as the father of modern epidemiology. Instead of looking for bad air, Snow suspected that the disease was being transmitted through contaminated water. He meticulously interviewed residents, plotting the exact addresses of cholera victims on a map of the affected area. His map quickly revealed a striking pattern. The vast majority of cholera deaths were clustered around a particular public water pump on Broad Street. Snow's investigation went further. He identified that a significant number of people who lived outside the immediate Broad Street area, but who frequented businesses or sent their children to schools near the pump, also contracted cholera. Conversely, he found that some residents living within the cluster did not get sick if they drew their water from a different, uncontaminated source. Or, famously, if they preferred to drink beer from a local brewery that had its own well. His most compelling evidence came from what he termed the grand experiment. He noted that two water companies supplied water to houses in the same London streets, the Lambeth Company and the Southwark and Vauxhall Company. Crucially, in 1852, the Lambeth Company had moved its water intake point upstream on the River Thames, above the polluted city, while Southwark and Vauxhall continued to draw water from the heavily contaminated lower Thames. By comparing mortality rates among households supplied by the two different companies, Snow found that those receiving water from Southwark and Vauxhall had a cholera death rate many times higher than those supplied by Lambeth. The houses were often side by side, creating a natural experiment where the only variable was the water source. Armed with this overwhelming evidence, Snow presented his findings to the local authorities, convincing them to remove the handle from the Broad Street pump. This decisive action, cutting off the source of contaminated water, brought the outbreak rapidly to an end. Snow concluded unequivocally that cholera was a waterborne disease, transmitted through contaminated water, and not by bad air. Neither Semmelweis nor Snow possessed the definitive proof of microorganisms. They could not see or culture the bacteria responsible for puerperal fever or cholera. The microscope of their era was not powerful enough to resolve these minuscule entities. However, their pioneering work provided the indispensable empirical foundation for the germ theory. Semmelweis demonstrated that an invisible agent could be transmitted from person to person or corpse to person via direct contact, and that simple hygiene could interrupt this transmission. Snow proved that another invisible agent could be transmitted through a common medium, contaminated water, and that removing the source of contamination could halt an epidemic. Their methods were groundbreaking. Semmelweis used quantitative data and a form of clinical trial. Snow used epidemiological mapping and statistical analysis. Both meticulously observed, rigorously hypothesized, and then intervened with practical solutions that saved lives. Their courage to challenge prevailing dogma, their reliance on observable facts, and their relentless pursuit of the truth, even when met with hostility, represent the true seeds of the germ theory. They showed what was happening and how to stop it, even if the why remained unseen. Their work created the intellectual climate and provided the compelling empirical evidence that would, just a few decades later, allow giants like Louis Pasteur and Robert Koch to finally identify and prove the existence of the microscopic world of pathogens, unleashing the full power of modern medicine against infectious diseases. The mid-19th century stood at a pivotal juncture in medicine. While pioneers like Semmelweis and Snow had provided compelling empirical evidence for the contagious nature of certain diseases and the role of environmental factors, the fundamental cause remained elusive. The dominant scientific paradigm still largely embraced the ancient concept of spontaneous generation, the belief that living organisms could arise de novo from non-living matter, and the miasma theory, which attributed disease to bad air. It was into this intellectual landscape that Louis Pasteur, a brilliant French chemist, stepped forth, not initially as a medical doctor, but as a meticulous experimentalist whose work would irrevocably dismantle these old theories and lay the irrefutable foundation for the germ theory of disease, thereby revolutionizing medicine. Pasteur's journey towards understanding disease began not in hospitals, but in breweries and vineyards. In the 1850s, the French beverage industry faced a severe crisis. Wine was turning sour, and beer was spoiling. Brewers and vintners sought a scientific explanation for these economically devastating phenomena. The prevailing belief was that these changes were purely chemical transformations, or that the tiny organisms found in spoiled liquids were simply products of spontaneous generation. Pasteur, however, suspected otherwise. Through a series of ingenious and rigorously designed experiments, Pasteur demonstrated that fermentation, the process by which grapes turn into wine and barley into beer, was not a purely chemical process, but a biological one. He proved that specific types of microscopic organisms, yeasts, were responsible for producing alcohol, while other, unwanted microorganisms, like bacteria, were responsible for turning the alcohol into vinegar or spoiling the beverages. He showed that if these unwanted microbes were prevented from entering the vats, the spoilage would not occur. His solution for preventing spoilage: heating the liquid to a specific temperature to kill the unwanted microorganisms, a process now universally known as pasteurization, saved the French wine and beer industries, and is still crucial to food safety today. This work on fermentation was his entry point into the world of microorganisms, but it also led him to challenge the cornerstone of prevailing biological thought: spontaneous generation. Proponents of spontaneous generation argued that if you left broth exposed to air, life would simply appear. To counter this, Pasteur designed his now famous swan neck flask experiment in 1861. He boiled broth in flasks with long, S-shaped necks. The air could enter the flask, but dust particles and microbes settled in the curve of the neck, unable to reach the sterile broth. The broth remained clear and sterile indefinitely. Only when the neck was broken, allowing dust and microbes to directly contact the broth, did contamination and microbial growth occur. This elegant experiment provided conclusive proof that microorganisms do not spontaneously generate, but arise only from pre-existing microorganisms, floating in the air. This groundbreaking demonstration shattered the theory of spontaneous generation, a fundamental intellectual barrier to understanding infectious disease. The implications of Pasteur's findings were profound. If microscopic organisms caused spoilage in wine, Could they not also cause disease in living beings? This was the pivotal leap from chemistry to medicine. Pasteur himself articulated this concept. The diseases of silkworms, the diseases of beer, the diseases of wine. These are all diseases having their origin in the invasion of a tiny organism, which is external to the host. This was the core idea of the germ theory of disease. That specific diseases are caused by specific, identifiable microorganisms. His next major medical triumph came in the 1860s, when he was called upon to investigate a devastating disease plaguing the French silk industry. Pébrine. Silkworms were dying in vast numbers, threatening a vital national industry. Through painstaking microscopic examination, Pasteur identified a parasitic microorganism as the cause of pébrine and developed methods to detect and eliminate infected silkworm eggs, effectively saving the industry. This success further reinforced his conviction. A specific microbe causes a specific disease. Pasteur's genius then extended to the development of vaccines, building upon the earlier empirical success of Jenner's smallpox vaccination. His breakthrough in this area came somewhat serendipitously. In 1879, while studying fowl cholera, Pasteur discovered that old, weakened cultures of the bacteria, when injected into chickens, did not cause disease, but instead protected the chickens from subsequent infection with virulent strains. He termed this weakened form attenuated, realizing that exposure to a weakened form of the pathogen could confer immunity without causing serious illness. This accidental discovery provided the scientific explanation for Jenner's earlier, intuitive work with cowpox and smallpox, providing the mechanism for vaccine development. Attenuation. He then applied this principle to anthrax, a deadly bacterial disease affecting livestock and sometimes humans. In 1881, Pasteur conducted a dramatic public experiment at Pouilly-le-Fort in France. He vaccinated a group of sheep with his attenuated anthrax vaccine, left another group unvaccinated, and then exposed both groups to virulent anthrax bacteria. The results were spectacular. All the vaccinated sheep survived, while all the unvaccinated sheep died. This public demonstration was a resounding triumph, solidifying the power of vaccination and confirming the germ theory on a grand scale. Pasteur's most celebrated achievement in vaccine development, and perhaps his most direct contribution to human medicine, was his rabies vaccine. Rabies was a terrifying and almost universally fatal disease, transmitted through the bite of an infected animal. In 1885, Pasteur, working with his assistants, developed a vaccine using dried spinal cord tissue from infected rabbits, which contained an attenuated form of the rabies virus. The first human recipient was a 9-year-old boy named Joseph Meister, who had been severely bitten by a rabid dog. Despite the immense ethical pressure and the fact that the rabies vaccine had only been tested on animals, Pasteur administered the vaccine. Joseph Meister survived, fully recovering, and lived a long life. This emotional and widely publicized success transformed Pasteur into a global hero and heralded the dawn of modern vaccinology, offering hope against previously untreatable diseases. Louis Pasteur's cumulative work provided the definitive, experimental proof for the germ theory of disease. He demonstrated, 1. That living organisms do not spontaneously arise. 2. That specific microorganisms cause specific processes, fermentation, and specific diseases, pebrine, fowl cholera, anthrax, rabies. 3. That these microorganisms can be manipulated to create attenuated forms that confer immunity, leading to effective vaccines. His rigorous experimental methodology and his irrefutable findings liberated medicine from the shackles of miasma and spontaneous generation. The germ theory transformed the understanding of infection, paving the way for Joseph Lister's antiseptic surgery, Robert Koch's isolation of specific pathogens, and the eventual development of antibiotics. It ushered in an era where diseases could be understood, diagnosed, prevented, and treated based on a scientific understanding of their microbial origins. Pasteur, the chemist, had, through his profound insights and tireless experimentation, fundamentally changed medicine forever, ushering in the age of microbiology and infectious disease control. While Louis Pasteur brilliantly established the conceptual framework of the germ theory of disease, demonstrating that invisible microorganisms cause fermentation and some diseases, he did not, for the most part, isolate the specific culprits responsible for human illnesses. The revolutionary step from a general theory of germs to the precise identification of which germ caused which disease, and how to definitively prove that link, belongs predominantly to the methodical and rigorous work of a German physician and bacteriologist, Robert Koch. Koch's contributions were not only about making groundbreaking discoveries, but also about developing the essential tools and scientific methodology that transformed bacteriology into a precise, systematic science, opening the floodgates for the identification of countless pathogens and laying the bedrock for modern medical diagnostics. Before Koch, even with Pasteur's insights, identifying the specific cause of an infection was akin to finding a needle in a haystack, an invisible needle in a biological haystack. A sick patient's body contained myriad bacteria, many of them harmless, some beneficial. How could one definitively prove that a particular microorganism was the sole cause of a specific disease? This was the monumental challenge that Koch set out to conquer. His genius lay in his relentless pursuit of definitive proof, leading him to formulate what became known as Koch's postulates, a set of 4 criteria that had to be met to establish a causal relationship between a microorganism and a disease. First articulated in 1882, these postulates provided an unparalleled standard of scientific rigor. 1. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. This means the suspected pathogen should be consistently present in sick individuals, but absent in healthy ones. 2. The microorganism must be isolated from a diseased organism and grown in pure culture. This was a critical challenge, requiring methods to separate the specific microbe from all others. 3. The cultured microorganism should cause disease when introduced into a healthy organism. This involved controlled experimentation, typically on laboratory animals, to recreate the disease. 4. The microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. This final step completed the logical chain of proof, ensuring the same microbe was responsible. These postulates, elegant in their simplicity, yet profound in their implications, provided the first truly scientific roadmap for attributing specific diseases to specific microbial agents. They became, and in modified forms still largely remain, the gold standard for establishing causality in infectious disease, demanding empirical evidence and reproducible results over theoretical speculation. But simply formulating the postulates wasn't enough. Koch also had to invent the means to fulfill them. The crucial technical challenge in isolating microorganisms was growing them in a pure culture. Until Koch, bacteria were typically grown in liquid broths, where different species would mix indiscriminately, making it impossible to study a single type. Koch's brilliant solution was to develop solid culture media. Initially, he used slices of potato or gelatin. Later, he revolutionized the field by introducing agar-agar as a solidifying agent, a substance derived from seaweed that remains solid at body temperature, unlike gelatin which melts. This innovation was transformative. By spreading a diluted sample of infected material onto a solid surface, individual bacterial cells would grow into distinct, visible colonies. Each colony would originate from a single bacterium, thus yielding a pure culture of that specific microorganism. This technique, further refined by his assistant Julius Richard Petri, who invented the shallow, round dish now universally known as the Petri dish, allowed for the systematic isolation, study, and identification of individual bacterial species. It enabled scientists to definitively separate the good bacteria from the bad, a feat previously impossible. Beyond pure cultures, Koch also pioneered the use of staining techniques. Microorganisms are often transparent and difficult to see under a microscope. Koch developed methods using aniline dyes, e.g., methylene blue, later the famous Gram stain, though developed by his contemporary Hans Christian Gram to color bacteria, making them visible and allowing for their differentiation based on their morphological characteristics and how they absorbed specific dyes. These tools, solid culture media, pure culture techniques, and staining methods were fundamental methodological breakthroughs that enabled bacteriology to blossom as a distinct scientific discipline. Armed with these revolutionary tools and his rigorous postulates, Koch embarked on a series of monumental discoveries that would forever alter the landscape of medicine. His first major triumph came in 1876, when he definitively proved that Bacillus anthracis was the causative agent of anthrax, a deadly disease affecting cattle, sheep, and occasionally humans. He successfully isolated the bacterium from infected animals, cultured it in pure form, injected it into healthy animals, which then developed anthrax, and re-isolated the same bacterium from the newly diseased animals. This was the first time that a bacterium had been unequivocally proven to cause a specific disease in accordance with his own rigorous criteria. It was a triumph that validated the germ theory in the most definitive way possible. However, Koch's most significant and globally impactful discovery came in 1882, when he announced the isolation of the bacterium responsible for tuberculosis, Mycobacterium tuberculosis. Tuberculosis, then known as consumption, was a devastating disease responsible for 1 in 7 deaths in Europe, striking down rich and poor alike, often with a slow, debilitating decline. Its cause had been a medical mystery for centuries. Koch's meticulous work, including his development of a specialized staining method, the acid-fast stain, to visualize this notoriously difficult-to-stain bacterium, culminated in his groundbreaking presentation on March 24, 1882. This discovery was met with immense excitement worldwide, as it identified the enemy behind one of humanity's deadliest scourges. It opened the door to understanding its transmission, developing diagnostic tests, and eventually, effective treatments and vaccines. For this monumental achievement, Robert Koch was awarded the Nobel Prize in Physiology or Medicine in 1905. Koch and his numerous brilliant students at his Institute for Infectious Diseases in Berlin went on to identify many other critical human pathogens, including the cholera Bacillus vibrio cholerae in 1883 during an epidemic in Egypt and later in India, building directly on John Snow's epidemiological insights. Other pathogens isolated by his students included the diphtheria bacillus, the typhoid bacillus, and the tetanus bacillus, among many others. Robert Koch's revolution was thus a revolution of precision and proof. He provided the definitive scientific framework, the practical tools, and the irrefutable evidence that fundamentally transformed medicine. His postulates demystified infectious diseases, moving them from the realm of vague miasmas or general contagions to specific, identifiable, and therefore targetable microbial entities. This shift directly paved the way for the development of accurate diagnostic tests, effective vaccines, antiseptic practices, as we will explore next with Lister, and ultimately, the age of antibiotics. Koch's systematic methodology became the cornerstone of infectious disease research, enabling scientists to wage a targeted war against pathogens and dramatically improve public health on a global scale. For centuries, the operating room was a place of last resort, a chamber of unimaginable suffering where surgery, a necessary evil, was often little more than a brutal race against time and agony. While the advent of anesthesia in the mid-19th century conquered pain, it paradoxically unmasked an even more formidable enemy: infection. Patients who survived the initial shock of the knife often succumbed days later to a horrifying array of postoperative complications: surgical sepsis, erysipelas, hospital gangrene, and cellulitis. Wounds that initially looked clean would inevitably fester, producing pus, odor, and eventually death, often through overwhelming systemic infection. The mortality rates for major surgeries, particularly amputations, were astronomically high, frequently exceeding 50 in the best hospitals. A surgeon's skill was measured by speed, not precision. For every second the patient lay exposed was another moment of agonizing, conscious suffering. What was unknown What remained invisible was the very cause of this rampant post-operative devastation. In the mid-19th century, the prevailing medical wisdom, the miasma theory, attributed these infections to bad air, or noxious effluvia, rising from decaying organic matter or sick patients themselves. Cleanliness was seen as generally beneficial, a moral virtue rather than a medical necessity. Surgeons would operate in their street clothes, often stained with blood and pus from previous procedures, considering such stains badges of honor or experience. Instruments were rarely sterilized, sponges were reused, and the operating theater itself was a breeding ground for unseen pathogens, a grim environment where patients literally breathed in the agents of their own demise. The sheer scale of death from infection made surgery a terrifying gamble, limiting its application to only the most desperate circumstances. The question loomed: how could surgery fulfill its promise if it was invariably followed by a hidden, deadly scourge? The answer, revolutionary in its simplicity, arrived through the profound insights of a British surgeon named Joseph Lister, 1827 to 1912, often hailed as the father of antiseptic surgery. Lister was a keen observer, deeply troubled by the rampant mortality in his surgical wards at the Glasgow Royal Infirmary. He noted, for example, the stark difference in outcomes between simple fractures, where the skin remained intact, and compound fractures, where the bone pierced the skin. Simple fractures usually healed without much complication, while compound fractures almost invariably led to a gruesome infection, often requiring amputation or proving fatal. This stark contrast suggested that exposure to the external environment was the critical factor, rather than some internal miasma arising from the patient himself. Lister's pivotal breakthrough came in the mid-1860s when he encountered the work of Louis Pasteur. Pasteur, as we've discussed, had conclusively demonstrated that fermentation and putrefaction, the process of decay, were caused by living microorganisms, or germs, present in the air, not by spontaneous generation. Lister, with a flash of profound insight, made the critical leap. If invisible germs caused putrefaction in organic matter, might they not also cause putrefaction infection in living tissues, specifically in open wounds? He hypothesized that infection was not due to some mysterious internal process or bad air from decay within the body, but rather from the entry of external microorganisms into the wound. This was a radical departure from accepted medical dogma. If germs were the enemy, then the solution was to destroy them or prevent their entry. Lister sought a chemical agent that could kill these microbes without causing excessive harm to the patient's tissues. His attention turned to carbolic acid phenol. He learned that carbolic acid was being used with success to treat sewage and eliminate the foul smell from a town in Carlisle, suggesting its potent antiseptic properties. In August 1865, he began his pioneering experiments. His first major success involved an 11-year-old boy with a compound fracture of the tibia, a notoriously lethal injury. Instead of immediately amputating, Lister meticulously cleaned the wound with a dressing soaked in carbolic acid, applied several layers of carbolized lint, and covered it with tin foil to prevent evaporation. the wound healed without suppuration or infection, a truly astonishing outcome for the time. Lister systematically extended his antiseptic methods to all aspects of surgery. He introduced the use of a carbolic acid spray in the operating room to create a germ-free atmosphere during operations, believing that the air itself was a source of contamination. He meticulously soaked surgical instruments, sponges, and even his own hands in dilute carbolic acid solutions before and during surgery. He also developed new antiseptic dressings and bandages saturated with carbolic acid to cover wounds postoperatively, creating a protective barrier against external germs. Furthermore, he revolutionized the material for surgical sutures. Recognizing that traditional silk sutures could harbor germs, he developed carbolized catgut sutures, which were absorbed by the body, removing the need for external removal and reducing the risk of infection. The results were dramatic and irrefutable. Lister's surgical mortality rates, particularly for major procedures like amputations and compound fractures, plummeted from over 45 to below 15, and in some cases, even to single digits. His wards, once notorious for hospital gangrene and the stench of infection, became remarkably clean and relatively odor-free. He presented his findings to the medical community, most notably in a series of articles in The Lancet in 1867, titled "On the Antiseptic Principle in the Practice of Surgery." Despite the overwhelming evidence and the undeniable improvement in patient outcomes, Lister's ideas faced considerable resistance and ridicule. His critics found the carbolic acid spray messy, smelly, and unpleasant. They scoffed at the idea of invisible germs and found the elaborate ritual of antiseptic preparation tedious and unnecessary, especially when they could not literally see the danger. His methods challenged deeply ingrained practices and the established authority of prominent surgeons who clung to the miasma theory. Many simply didn't believe in the germs he spoke of. However, Lister, a man of quiet conviction, persisted. He traveled extensively, demonstrating his techniques and presenting his compelling data. Gradually, as more surgeons adopted his methods and saw the dramatic reduction in deaths with their own eyes, acceptance grew. His tour of Germany and the United States in the 1870s proved particularly influential, as his techniques were embraced by pioneering surgeons there. Lister's legacy is immense and foundational. His antiseptic principle, born from the rigorous application of Pasteur's germ theory, transformed surgery from a perilous gamble into a life-saving science. By proving that infections were caused by external, specific microorganisms, he ushered in an era where infection could be actively prevented and controlled. His work paved the way for the eventual transition from antiseptic surgery killing germs to aseptic surgery, preventing germs from entering the surgical field in the first place. This led to the development of the sterile operating room, with sterilized instruments, drapes, gowns, masks, and gloves, the practices that are now standard across the globe. The ability to operate in a clean, infection-free environment liberated surgeons to perform complex internal procedures that were previously unthinkable. Anesthesia had solved the problem of pain. Lister solved the problem of infection. Together, these two revolutions fundamentally redefined surgery, allowing for operations on the brain, heart, and abdominal organs, thereby saving countless lives and fundamentally altering the trajectory of medical advancement. Joseph Lister's tireless dedication to applying scientific principles to clinical practice not only reduced immense suffering and death, but also solidified the germ theory's acceptance and made modern surgery, as we know it today, a reality. His impact reverberates in every sterile instrument, every gloved hand, and every successful surgical outcome across the world. Even as the germ theory of disease was beginning to unravel the mysteries of infection, a parallel transformation was underway in two other critical areas of medicine: the development of therapeutic agents, or drugs, and the ability to non-invasively visualize the body's interior. For centuries, the practice of medicine had relied on a pharmacopoeia of remedies derived from natural sources: plants, minerals, animal products, whose efficacy was often based on folklore, anecdotal evidence, or outright mysticism. Diagnosis, similarly, was largely a blind art, relying on external symptoms, palpation, and educated guesswork to infer what lay hidden beneath the skin. The late 19th and early 20th centuries witnessed a dramatic shift from these empirical, often mystical approaches to a scientific, mechanistic understanding of how drugs affect the body and how internal structures could be revealed, laying the groundwork for modern pharmacology and diagnostic imaging. For most of history, medical treatments consisted of complex concoctions of herbs, often prescribed based on ancient traditions. The doctrine of signatures, where a plant's appearance hinted at its use, or the prevailing humoral theories. Potency varied wildly, active ingredients were unknown, and efficacy was inconsistent at best. The shift towards a mechanistic pharmacology began with the isolation of pure, active chemical compounds from these traditional remedies. The goal was to identify the specific substance responsible for the therapeutic effect, understand its chemical nature, and standardize its dosage. One of the earliest and most impactful breakthroughs in this area came in 1804, when the German pharmacist Friedrich Sertürner successfully isolated morphine from opium. For centuries, opium, derived from the poppy plant, had been known for its powerful pain-relieving and sedative properties. Sertürner's achievement marked the first isolation of an active ingredient from a plant, demonstrating that a single chemical compound, not the whole plant, was responsible for its potent effects. This opened up the entire field of alkaloid chemistry and provided physicians with a powerful, standardized painkiller, revolutionizing pain management, particularly after surgical procedures. Following Sertürner's lead, other chemists began to isolate active compounds from other traditional remedies. In 1820, French chemists Pierre-Joseph Pelletier and Joseph Bien-Aimé Caventou isolated quinine from cinchona bark, a substance known since the 17th century to treat malaria. Quinine became the first effective specific treatment for a major infectious disease, saving countless lives in malaria-ridden regions worldwide. These successes heralded a new era. Medicine could now wield specific chemical bullets against specific ailments, moving away from generalized tonics and purges. The ultimate triumph of this mechanistic approach was not just the isolation of natural compounds, but the ability to synthesize new chemical compounds in the laboratory, designed specifically for therapeutic purposes. This marked the birth of modern pharmaceutical chemistry. A prime example is the development of aspirin. For centuries, preparations from willow bark had been used to relieve pain and fever. In the mid-19th century, salicylic acid, the active ingredient in willow bark, was isolated and later synthesized. However, it caused severe stomach irritation. It was Felix Hoffmann, a chemist working for Bayer in Germany, who, in 1897, successfully synthesized a less irritating derivative, acetylsalicylic acid, which Bayer marketed as aspirin. Aspirin quickly became the world's most widely used drug, a cornerstone of pain relief, fever reduction, and anti-inflammation, and later recognized for its antiplatelet properties. Its development showcased how chemical modification could enhance efficacy and reduce side effects, demonstrating a true mechanistic understanding. Perhaps the most iconic representation of this new magic bullet concept in early pharmacology came with the work of Paul Ehrlich, 1854 to 1915. A German physician and scientist, Ehrlich was deeply influenced by the germ theory and believed that it should be possible to find chemical compounds that would selectively kill disease-causing microorganisms without harming the host. He coined the term chemotherapy to describe this targeted approach. His tireless research, involving thousands of experiments with various dyes and chemical compounds, aimed to find such a selective poison. His ultimate triumph came in 1909 with the synthesis of salvarsan arsphenomen. This arsenic-based compound proved remarkably effective in treating syphilis, a widespread and devastating venereal disease. Salvarsan was the first truly synthetic drug designed to specifically target a pathogen within the body, marking a monumental step in the direct application of chemistry to infectious disease treatment. It was an imperfect drug with significant side effects, but it validated Ehrlich's magic bullet concept and set the stage for the search for even safer and more effective antimicrobial agents, a quest that would ultimately lead to antibiotics. Concurrent with these revolutionary developments in pharmacology, an equally profound breakthrough occurred in diagnostics, forever changing how physicians could peer inside the human body without resorting to surgery. Before the late 19th century, diagnosing internal conditions was largely based on external signs: palpation, feeling with hands, percussion tapping to listen for sounds, auscultation, listening with a stethoscope, and the patient's reported symptoms. To confirm internal injuries or diseases, exploratory surgery was often the only option, a dangerous and often fatal proposition before modern anesthesia and aseptic techniques. This era of diagnostic blindness was shattered on November 8, 1895, by the serendipitous discovery of Wilhelm Conrad Röntgen, 1845 to 1923, a German physicist. While experimenting with cathode ray tubes in his laboratory, Röntgen noticed a faint glow emanating from a fluorescent screen nearby, even when the tube was completely covered. He realized that a new, invisible kind of ray was being emitted, capable of passing through opaque objects. He dubbed these mysterious emanations X-rays because their nature was unknown. The true significance of his discovery became apparent when he placed his wife Bertha's hand between the cathode ray tube and a photographic plate. To their astonishment, the developed plate revealed a clear image of the bones within her hand, along with her wedding ring. This was the first X-ray image, a ghostly yet strikingly clear window into the living body. The impact of X-rays on medicine was immediate and profound. Within months of Röntgen's announcement, hospitals across the globe acquired X-ray machines. For the first time, physicians could non-invasively visualize internal structures. Orthopedics, fractures, dislocations, and bone deformities, once diagnosed by touch and guesswork, could now be seen with unprecedented clarity. This revolutionized the setting of bones and the monitoring of healing. Surgery. Surgeons could precisely locate bullets, needles, or other foreign objects within the body, allowing for more targeted and safer surgical interventions. Pathology. The ability to visualize lung lesions in tuberculosis patients provided early diagnostic capability for this widespread killer. Later, with the introduction of contrast agents, internal organs like the gastrointestinal tract and blood vessels could also be visualized. The discovery of X-rays was a leap from external observation to internal structural revelation. It provided an objective, visual basis for diagnosis, reducing uncertainty and allowing for more informed treatment decisions. It cemented the growing belief that disease had a definable, physical basis that could be seen and understood. While the long-term dangers of radiation exposure were not initially understood, leading to early abuses like shoe-fitting fluoroscopes and radiation burns, the revolutionary power of X-rays quickly made them an indispensable diagnostic tool. In essence, the move from mysticism to mechanism in early pharmacology and X-ray diagnostics represents a critical phase in the evolution of modern medicine. The ability to identify, isolate, and synthesize specific chemical compounds for targeted therapeutic action, exemplified by aspirin and salvarsan, transformed drug development from an art of folklore into a science of chemistry. Simultaneously, Röntgen's discovery of X-rays provided an unprecedented, non-invasive window into the body's hidden architecture, revolutionizing diagnosis and guiding surgical interventions with a clarity previously unimaginable. Together, these advancements moved medicine definitively into an era of scientific precision, mechanistic understanding, and technologically empowered diagnosis and treatment. For countless millennia, humanity endured a brutal existence under the dominion of microscopic adversaries. Bacterial infections were the silent, relentless killers, shaping demographics, ending lives prematurely, and dictating the very course of civilization. A scraped knee could turn into fatal sepsis. A common cold could descend into deadly pneumonia. And even childbirth, a moment of profound joy, was shadowed by the ever-present specter of childbed fever. Before the mid-20th century, there was little effective defense against these microbial onslaughts. Treatments were often symptomatic, relying on the body's own strength, or employing desperate measures like bloodletting, purges, or crude herbal remedies with uncertain efficacy. The germ theory, definitively proven by Pasteur and Koch, had identified the enemy, but the magic bullet to slay them remained elusive. This terrifying vulnerability to bacterial infections made life precarious, surgery hazardous, and minor illnesses potentially lethal. Then, almost by serendipity, a revolution began, one that would redefine medicine and save hundreds of millions of lives: the discovery and development of antibiotics. The story begins in 1928, in a rather cluttered laboratory at St. Mary's Hospital in London, belonging to a Scottish bacteriologist named Alexander Fleming, 1881 to 1955. Fleming was known for being a somewhat untidy scientist, often leaving bacterial cultures exposed. Returning from a summer holiday, he noticed something peculiar on a Petri dish containing Staphylococcus bacteria, a common cause of infections. A patch of blue-green mold, identified as Penicillium notatum, had contaminated the dish. Crucially, around this mold, there was a clear ring, a zone of inhibition, where the Staphylococcus bacteria had failed to grow. Fleming, with a keen eye for the unusual, immediately recognized the significance of this observation. The mold was producing something that inhibited bacterial growth. He cultivated the mold, grew it in liquid culture, and confirmed that the mold juice, as he called it, was indeed capable of killing a wide range of pathogenic bacteria, including those responsible for scarlet fever, pneumonia, meningitis, and diphtheria. He named the active substance penicillin after the mold from which it originated. In 1929, he published his findings in the British Journal of Experimental Pathology. However, Fleming struggled to isolate and purify the unstable compound in sufficient quantities for practical use. He also found it challenging to keep the substance active for long, and it seemed to work best in a test tube, not always in living organisms. Concluding that penicillin was too difficult to produce on a large scale and too unstable to be a practical treatment, he essentially set the discovery aside, though he continued to use it for laboratory purposes to selectively inhibit certain bacteria. For over a decade, penicillin remained a scientific curiosity, a promising but unfulfilled potential. The true miracle of penicillin, its transformation from a laboratory curiosity into a life-saving drug, required the efforts of another brilliant team. This occurred more than a decade later, during the ominous shadow of World War II, at the University of Oxford. In 1939, a German Jewish refugee biochemist, Ernst Chain 1906 to 1979, was reviewing scientific literature when he stumbled upon Fleming's overlooked paper on penicillin. Intrigued, he brought it to the attention of his supervisor, Howard Florey, 1898 to 1968, an Australian pharmacologist and pathologist who headed the Sir William Dunn School of Pathology. Together with their brilliant and pragmatic colleague, Norman Heatley, 1911 to 2004, they embarked on the arduous task of purifying penicillin and testing its therapeutic potential. The Oxford team faced immense challenges. Penicillin was incredibly difficult to purify and was produced in minuscule amounts by the mold. it was notoriously unstable, quickly degrading. They developed ingenious methods for extracting and concentrating the elusive compound, often resorting to crude but effective techniques. They conducted systematic animal experiments, inoculating mice with lethal doses of streptococci, and then treating them with penicillin. The results were astounding. The untreated mice died, while the penicillin-treated mice survived. This incontrovertible evidence of its efficacy in living organisms fueled their determination. The definitive moment arrived on February 12, 1941, when they administered penicillin to their first human patient, Albert Alexander, a London policeman suffering from a severe staphylococcal and streptococcal infection that had caused abscesses and boils all over his face and body. The penicillin initially worked wonders, visibly clearing the infection. However, the supply of the drug was so meager that, when it ran out, Alexander tragically relapsed and died. This heartbreaking setback only intensified the team's resolve. They knew penicillin worked, but they needed to produce it on an industrial scale. Recognizing that wartime Britain lacked the resources for mass production, Florey and Heatley traveled to the United States in 1941, seeking collaboration. With the support of the U.S. government and American pharmaceutical companies, a concerted effort was launched, bringing together chemists, engineers, and microbiologists in a frantic race against time. Scientists at the Peoria, Illinois lab of the U.S. Department of Agriculture discovered a new strain of Penicillium chrysogenum that produced far more penicillin and developed methods for deep tank fermentation. Pharmaceutical companies rapidly scaled up production. By the D-Day invasion in June 1944, enough penicillin was being produced to treat all Allied casualties, dramatically reducing deaths from battlefield infections, which had historically killed more soldiers than combat injuries themselves. The impact of penicillin was nothing short of miraculous. Diseases that were once death sentences, pneumonia, syphilis, gonorrhea, diphtheria, meningitis, osteomyelitis, suddenly became treatable. Child mortality plummeted. Surgical outcomes improved dramatically as postoperative infections, once rampant, could now be controlled. The average human lifespan increased significantly. In 1945, Alexander Fleming, Howard Florey, and Ernst Chain were jointly awarded the Nobel Prize in Physiology or Medicine for their groundbreaking work, forever cementing penicillin's place in history as the world's first true miracle drug. The success of penicillin spurred an intense global search for new antibiotics, ushering in what is often called the golden age of antibiotics in the mid-20th century. Scientists screened thousands of soil samples from around the world, recognizing that microorganisms in the soil produced compounds to compete with each other. This led to the discovery of other classes of antibiotics. Streptomycin, discovered by Selman Waxman and his team in 1943, it was the first effective drug against tuberculosis, a disease that had plagued humanity for millennia. Tetracyclines, broad-spectrum antibiotics discovered in the late 1940s, effective against a wide range of bacterial infections. Chloramphenicol, erythromycin, neomycin, vancomycin. Each new discovery broadened the arsenal against bacterial pathogens, transforming healthcare. For decades, it seemed humanity had won the war against bacteria. The ability to cure once fatal infections became almost routine. However, this triumph inadvertently led to complacency and ultimately a new and terrifying challenge: antibiotic resistance. The very success of antibiotics created an evolutionary pressure. Bacteria, being incredibly adaptable and fast-reproducing organisms, developed mechanisms to resist the drugs designed to kill them. This phenomenon, known as antibiotic resistance, is driven by the overuse and misuse of antibiotics in human medicine, agriculture, and livestock. Every time an antibiotic is used, it kills susceptible bacteria, leaving behind the few resistant ones to multiply, passing on their resistance genes. Over time, these resistant strains become dominant, rendering our most powerful drugs ineffective. The emergence of superbugs, like methicillin-resistant Staphylococcus aureus, MRSA, vancomycin-resistant Enterococci VRE, and drug-resistant tuberculosis, is a global health crisis. Infections that were once easily treatable are now becoming untreatable, pushing medicine back to a pre-antibiotic era for some patients. The pipeline for new antibiotic discovery has slowed significantly in recent decades, making the challenge even more acute. Today, the miracle of antibiotics is intertwined with the urgent need for antibiotic stewardship, the responsible use of these precious drugs to preserve their effectiveness. This involves prescribing antibiotics only when necessary, completing full courses of treatment, reducing their use in agriculture, and investing in new drug discovery. The discovery of penicillin and the subsequent development of other antibiotics represent one of the greatest achievements in medical history, fundamentally changing life expectancy and the practice of medicine. They transformed deadly infections into manageable illnesses. However, the ongoing fight against antibiotic resistance reminds us that the battle against microorganisms is a continuous, evolving challenge, demanding vigilance, innovation, and global collaboration to ensure that the miracle of antibiotics endures for future generations. For much of medical history, the human body was largely understood through the lens of anatomy, its visible structures. And, as the 19th century progressed, through the recognition of external infectious agents. Yet a vast array of debilitating conditions persisted, mysterious and unyielding, seemingly unrelated to trauma or invading microbes. Conditions like diabetes, goiter, rickets, beriberi, and scurvy plagued populations, baffling physicians and leading to chronic suffering and death. The dramatic breakthroughs in the late 19th and early 20th centuries that unraveled the secrets of hormones and vitamins fundamentally shifted this understanding. They revealed a hidden universe of internal chemical messengers and essential micronutrients, transforming medicine from a focus primarily on external threats to an appreciation of the body's intricate internal biochemistry and the profound impact of diet on health. This marked the birth of endocrinology and modern nutritional science. The concept of hormones emerged from a growing understanding that organs could influence each other not just through nerves, but through chemical signals transported via the bloodstream. For centuries, observations had hinted at this. The effects of castration on men and animals, the dramatic physical and mental changes associated with thyroid disorders, or the mysterious wasting disease of diabetes, all pointed to some form of internal regulation. However, the precise mechanisms remained obscure. The definitive birth of endocrinology, the study of hormones, can be traced to 1902 British physiologists William Bayliss and Ernest Starling, while studying digestion, discovered that a substance produced in the duodenum, upon contact with stomach acid, stimulated the pancreas to secrete digestive juices. They isolated this chemical messenger and named it secretin. Starling later coined the term hormone from the Greek hormao, meaning I arouse, or set in motion, in 1905 to describe such chemical messengers that are produced in one part of the body and transported by the bloodstream to act on distant target organs. This discovery established a revolutionary principle. The body communicated and regulated its functions through a complex network of chemical signals. Following secretin, the floodgates opened. In 1904, the first hormone to be isolated and synthesized was adrenaline epinephrine by a Japanese chemist, Jikichi Takamine, and an American pharmacologist, Thomas Aldrich. This powerful substance, produced by the adrenal glands, was quickly recognized for its role in the fight or flight response, affecting heart rate, blood pressure, and metabolism. Its isolation provided a pure, standardized chemical that could be administered therapeutically, proving the immense potential of hormone replacement. However, the most dramatic and life-saving discovery in this field was undoubtedly insulin. For millennia, type 1 diabetes was a relentless and fatal disease, particularly in children. Sufferers would slowly waste away, starved of the ability to utilize glucose, despite consuming adequate food. Physicians could offer little more than dietary restriction, prolonging life by only a few painful months or years. The pancreas was suspected to play a role, but the specific chemical factor it produced remained elusive. In 1921, a young Canadian surgeon, Frederick Banting, along with his medical student assistant, Charles Best, working under the guidance of Professor John MacLeod, and with the critical biochemical expertise of James Collip, embarked on a series of experiments at the University of Toronto. Through painstaking work involving ligation of pancreatic ducts in dogs to destroy digestive enzymes, and then extracting a substance from the degenerated pancreas, Banting and Best isolated a crude extract that lowered blood sugar in diabetic dogs. Collip then refined this extract, purifying it sufficiently for human use. In January 1922, Leonard Thompson, a 14-year-old boy dying of diabetes in a Toronto hospital, became the first human recipient of insulin. The initial crude extract caused an allergic reaction, but a purified version, administered a few days later, dramatically lowered his dangerously high blood sugar. The effect was astonishing, literally pulling him back from the brink of death. The discovery of insulin was an immediate medical sensation. Within a few years, large-scale production methods were developed, making insulin widely available. It transformed type 1 diabetes from a death sentence into a manageable chronic condition, allowing millions of people to lead full, productive lives. Banting and MacLeod were awarded the Nobel Prize in Physiology or Medicine in 1923, an honor they controversially shared with Best and Collip, recognizing the collaborative nature of this monumental discovery. Insulin's success firmly established the power of hormone replacement therapy and ignited the search for other endocrine disorders and their potential chemical remedies, leading to the development of treatments for thyroid deficiencies, e.g., thyroxine for myxedema, and a deeper understanding of adrenal, pituitary, and sex hormones. Parallel to the unfolding story of hormones was the groundbreaking revelation of vitamins, a class of organic compounds required in minute quantities for normal growth and metabolism, which the body cannot synthesize on its own. For centuries, specific diseases had afflicted populations, seemingly at random, yet often linked to specific diets or living conditions. Scurvy ravaged sailors on long voyages. Beriberi paralyzed populations reliant on polished rice. Rickets deformed children in industrial cities, and pellagra caused dermatitis, diarrhea, and dementia in impoverished communities. These were not infectious diseases. Their cause was a profound mystery. The first crucial clues came from observing these deficiency diseases. In the late 19th century, Christian Eichmann, a Dutch physician working in the Dutch East Indies, now Indonesia, observed that chickens fed polished rice developed symptoms strikingly similar to beriberi, while those fed unpolished rice did not. He initially hypothesized a toxin in polished rice, but later concluded that there was an essential protective factor present in the rice bran that was removed during polishing. Though he didn't identify the specific chemical, his work provided powerful evidence for the existence of accessory food factors. Inspired by Eichmann's work, the English biochemist Frederick Gowland Hopkins, in 1906, hypothesized the existence of accessory food factors beyond proteins, fats, and carbohydrates, which were crucial for health. His animal experiments demonstrated that a diet of pure casein, fat, carbohydrates, and minerals could not sustain life, but adding small amounts of milk or yeast allowed the animals to thrive. He argued that these accessory factors, though present in tiny amounts, were essential for preventing deficiency diseases. The term vitamine, originally with an E for amine, as it was thought they were all amines, was coined in 1912 by Casimir Funk, a Polish biochemist, when he isolated a factor from rice bran that cured beriberi, which we now know as thiamine vitamin B1. Although not all vitamins turned out to be amines, the name stuck, with the E later dropped. The 20th century witnessed a rapid succession of vitamin discoveries. Vitamin C, ascorbic acid. The cure for scurvy, known empirically for centuries. James Linn's work in the 18th century showed citrus cured scurvy, was finally isolated in 1928 by Albert Szent-Györgyi. Vitamin D, identified as the antirachitic factor in the 1920s, solving the mystery of rickets, a bone-deforming disease prevalent in children in sun-deprived industrial cities. Its link to sunlight exposure and dietary intake of certain fats was established. Vitamin A, B complex beyond B1, K, E. Each discovery revealed another piece of the intricate puzzle of human metabolism and provided a specific remedy for a previously baffling ailment. The recognition and isolation of vitamins revolutionized nutritional science. It moved the focus beyond simply consuming enough calories and macronutrients, proteins, fats, carbohydrates, to understanding the critical importance of micronutrients. This led to public health campaigns. Governments and health organizations initiated campaigns to encourage the consumption of vitamin-rich foods or to fortify staple foods, e.g., adding iodine to salt to prevent goiter, vitamin D to milk, vitamin B to grains. Eradication of deficiency diseases. Diseases like scurvy, rickets, beriberi, and pellagra, once scourges, became rare in populations with access to diverse diets or fortified foods. A deeper understanding of metabolism. Vitamins were found to act as coenzymes, essential for various metabolic processes, integrating nutrition directly into biochemistry. In conclusion, the discoveries of hormones and vitamins were twin revolutions that fundamentally transformed the understanding of disease. They revealed that the human body was not merely a collection of organs susceptible to external threats, but an exquisitely complex chemical factory, regulated by subtle internal messengers, and absolutely dependent on trace amounts of essential compounds from the diet. This shift from a purely anatomical or infection-centric view to a biochemical and physiological one opened entirely new avenues for diagnosis, prevention, and treatment. It led to life-saving therapies for conditions like diabetes and thyroid disorders, and the virtual eradication of many devastating deficiency diseases. These profound insights into the body's internal workings and its nutritional needs cemented the role of biochemistry and nutritional science as indispensable pillars of modern medicine, extending its reach from reactive treatment to proactive health maintenance and the optimization of human well-being. While individual medical breakthroughs, the invention of anesthesia, the discovery of antibiotics rightly capture our imagination for their dramatic impact on treating the sick. A silent, equally profound revolution has unfolded over centuries, one that has arguably saved more lives and prevented more suffering than any other aspect of medicine: the relentless advancement of public health. This shift in focus from treating the individual patient to safeguarding the health of entire populations through organized efforts in sanitation, widespread vaccination, and the systematic study of disease patterns epidemiology, stands as a cornerstone of modern medicine and a testament to collective human ingenuity. For millennia, the health of populations was at the mercy of chaotic forces. Cities were often cesspools, breeding grounds for disease, and epidemics swept through communities with terrifying regularity. The very concept of public health as a deliberate, organized endeavor was embryonic. While ancient Rome boasted impressive aqueducts and sewers, these were exceptions, often lost during the decline of empires. The Industrial Revolution of the 18th and 19th centuries, with its rapid urbanization and overcrowding, exacerbated these problems, leading to unprecedented levels of filth, squalor, and the rampant spread of infectious diseases like cholera, typhoid, and tuberculosis. These were the grim realities that spurred the modern public health movement. The first great public health triumph of the modern era was the sanitation revolution. Even before the full acceptance of germ theory, far-sighted reformers understood that there was a powerful, if mysterious, link between environmental filth and disease. Edwin Chadwick, a social reformer in Victorian England, was a leading proponent of the sanitary idea. Though initially influenced by the miasma theory, he passionately argued for government intervention to improve the living conditions of the poor. His influential 1842 report, "The Sanitary Condition of the Laboring Population of Great Britain," vividly documented the horrific conditions in slums and directly linked them to high rates of disease and premature death. He advocated for clean water, efficient sewage systems, and proper waste disposal as essential measures to combat illness. Perhaps the most iconic achievement of this early sanitation movement was the construction of London's vast modern sewer system. In the scorching summer of 1858, the great stink of the River Thames, overwhelmed by raw sewage, became so unbearable that it crippled parliamentary proceedings. This immediate crisis, combined with the growing understanding that cholera outbreaks were linked to polluted water, thanks to John Snow's epidemiological work, which we will revisit, spurred action. Joseph Basil Jett, a brilliant civil engineer, designed and oversaw the construction of a monumental underground network of intercepting sewers and pumping stations, diverting London's waste away from the Thames. This audacious project, completed over many years, dramatically reduced the incidence of waterborne diseases in London, proving the immense power of large-scale infrastructure in improving public health. Similar initiatives followed in other major cities around the world, leading to widespread improvements in clean water provision, sewage treatment, and waste management. The eventual scientific confirmation of the germ theory by Pasteur and Koch provided the definitive why for these empirical successes. It revealed that the unseen pathogens thriving in contaminated water and sewage were the true agents of disease, thus reinforcing the vital importance of sanitation beyond simply removing bad smells. The provision of clean drinking water and effective waste disposal became, and remains, fundamental pillars of public health globally, responsible for a significant proportion of the increase in life expectancy over the last 150 years. Hand in hand with sanitation, the systematic development and deployment of vaccinations launched a proactive offensive against infectious diseases, transforming them from inevitable scourges into preventable conditions. Building on Edward Jenner's pioneering work with smallpox and Louis Pasteur's conceptualization of attenuated vaccines, the late 19th and 20th centuries witnessed an explosion in vaccine development. Once the specific bacterial and viral culprits of diseases were identified, thanks largely to Koch, scientists could begin to develop targeted vaccines. Pasteur himself continued to lead the way with his rabies vaccine. The early 20th century saw the development of vaccines for diphtheria, tetanus, and typhoid. The mid-20th century, however, marked another golden age of vaccinology, particularly against devastating childhood diseases. Polio. The development of the inactivated polio vaccine by Jonas Salk in 1955, followed by the oral attenuated vaccine by Albert Sabin in 1961, effectively brought an end to the terrifying epidemics of poliomyelitis that caused paralysis and death in children worldwide. Mass vaccination campaigns in the 1950s and 60s virtually eliminated the disease in developed nations. Measles, mumps, and rubella, MMR. The development of combined vaccines dramatically reduced the incidence of these common childhood illnesses, preventing complications like encephalitis, deafness, and congenital rubella syndrome. Whooping cough pertussis, hepatitis B, Haemophilus influenzae type B Hib, pneumococcal disease. One by one, vaccines emerged to protect against these pathogens, collectively saving millions of lives and preventing immense suffering and disability. The concept of herd immunity became central to vaccination strategies. This principle recognizes that when a sufficiently high percentage of a population is immune to a disease, either through vaccination or prior infection, it becomes difficult for the disease to spread, thereby protecting even those who cannot be vaccinated. E.g., infants, immunocompromised individuals. Global vaccination campaigns, particularly the monumental effort to eradicate smallpox led by the World Health Organization, showcased the power of this strategy. Through tireless, coordinated efforts to vaccinate populations worldwide, often under challenging conditions, smallpox was declared officially eradicated in 1980, the only human disease ever to be completely wiped out. This stands as a powerful testament to the collective will and scientific potential unleashed by vaccination. Vaccinations have not only prevented countless deaths, but have also had profound economic and societal benefits, reducing healthcare burdens and allowing children to grow into healthy, productive adults. While new challenges like vaccine hesitancy and the emergence of novel pathogens, as seen with the rapid development of COVID-19 vaccines, persist, vaccination remains one of the most cost-effective and impactful public health interventions in history. The third pillar of advancing public health, without which systematic sanitation and vaccination efforts would be less effective, is epidemiology. Building directly upon the empirical work of John Snow in the 19th century, epidemiology evolved into a formal scientific discipline in the 20th century. John Snow, through his meticulous mapping of cholera cases and identification of the Broad Street pump, laid the methodological groundwork for modern epidemiology. The systematic study of the distribution and determinants of health-related states or events in specified populations, and the application of this study to the control of health problems. With the advent of germ theory, epidemiology gained a powerful explanatory framework. Epidemiologists could now identify specific pathogens, trace their pathways of transmission, identify risk factors, and design targeted interventions. The 20th century saw epidemiology expand its scope far beyond infectious diseases to investigate chronic conditions like heart disease, cancer, and diabetes, identifying crucial risk factors such as smoking, diet, and lack of exercise. Modern epidemiology employs sophisticated statistical methods, surveillance systems, and robust study designs, e.g., cohort studies, case control studies too. Investigate outbreaks. Rapidly identify the source and mode of transmission of new or re-emerging diseases. Identify risk factors. Uncover the behaviors, environmental exposures, or genetic predispositions that increase the likelihood of disease. Evaluate interventions. Assess the effectiveness of new drugs, vaccines, or public health policies. Inform public policy. Provide the evidence base for decisions on everything from food safety regulations to air quality standards and health education campaigns. The critical role of epidemiology was vividly demonstrated during the COVID-19 pandemic, where epidemiologists worldwide tracked the virus's spread, identified transmission routes, evaluated vaccine efficacy, and guided public health responses, showcasing its indispensable role in global health security. In conclusion, the advancements in public health through sanitation, vaccinations, and epidemiology represent a profound and ongoing evolution in medicine. They mark a strategic shift from a reactive, individualistic approach to a proactive, population-level one. Sanitation transformed urban environments, making them healthier and more habitable. Vaccinations provided the means to prevent devastating infectious diseases, altering global disease patterns and increasing life expectancy. Epidemiology provided the scientific rigor to understand disease patterns, identify causes, and guide effective interventions. Together, these three pillars form the formidable foundation of modern public health, continually working to prevent disease, promote health, and improve the quality of life for billions of people worldwide, truly reflecting medicine's highest ambition: to keep populations healthy, not just to cure individuals. War, in its brutal efficiency, has paradoxically been one of the most powerful and relentless drivers of medical advancement throughout history. From the crude field dressings of ancient armies to the sophisticated trauma centers and blood banks of today, the sheer scale and severity of injuries inflicted during conflict have compelled physicians, surgeons, and scientists to innovate, often at an accelerated pace, unmatched in times of peace. The urgency of saving lives on the battlefield has pushed the boundaries of surgical technique, spurred breakthroughs in infection control, and perhaps most profoundly, revolutionized our understanding and practice of transfusion medicine. Even in ancient times, the exigencies of warfare led to early medical organization. The Romans, as we've explored, established valetudinaria, dedicated military hospitals, and developed rudimentary surgical instruments for battlefield trauma. Throughout subsequent centuries, military campaigns, sieges, and battles provided grim laboratories for surgeons to hone their skills in amputation, wound management, and basic triage. Yet, for much of this history, the wounded soldier faced a terrifying prognosis: pain beyond imagination, a high likelihood of agonizing infection, and a limited chance of survival, particularly from severe hemorrhage or internal injuries. The true acceleration of medical progress driven by conflict began in the 19th century and intensified dramatically in the 20th century. The American Civil War 1861 to 1865, for instance, despite its horrific casualty rates, played a pivotal role in the establishment of organized battlefield triage, ambulance systems for rapid evacuation, and the formation of the U.S. Army Medical Museum, which meticulously collected pathological specimens, advancing the understanding of war wounds. The sheer volume of gunshot wounds, particularly from the new, more destructive Mini-A ball, forced surgeons to confront complex fractures and massive tissue damage, leading to improvements in wound care and surgical techniques, though infection remained a deadly adversary. The First World War 1914 to 1918 presented unprecedented challenges. The nature of trench warfare and the advent of high-explosive shells and machine guns led to a new type of devastating injury. Complex, contaminated wounds with extensive tissue damage and multiple fractures. This era saw the widespread adoption of antiseptic techniques, building on Lister's principles, to combat the rampant infections that had plagued earlier conflicts. Surgeons learned to debride clean-out wounds more effectively, and the use of continuous irrigation with antiseptic solutions, such as Dakin's solution, became common. The war also accelerated the development of orthopedics and reconstructive surgery, as doctors grappled with salvaging limbs and repairing mutilated faces. New methods for splinting complex fractures, particularly the Thomas splint, significantly reduced mortality from femoral fractures, once almost invariably fatal. The need for comprehensive care for the maimed and shell-shocked also led to nascent fields of physical therapy, occupational therapy, and mental health support. However, a persistent and life-threatening problem remained: hemorrhagic shock. Soldiers bleeding out from massive trauma faced near-certain death, often before reaching definitive medical care. The challenge was how to replace lost blood volume quickly and effectively. This necessity gave birth to transfusion medicine, arguably one of war's most enduring and life-saving legacies. The concept of transferring blood from one individual to another had fascinated physicians for centuries, but early attempts were almost universally disastrous. As early as the 17th century, some crude animal-to-human transfusions were attempted, but these often resulted in fatal reactions, without any understanding of why. Direct human-to-human transfusions, though occasionally successful by sheer luck, were equally perilous. The key to safe blood transfusion remained hidden until the dawn of the 20th century. The monumental breakthrough came in 1900 with the work of Austrian physician Karl Landsteiner, 1868 to 1943. Landsteiner discovered the ABO blood groups. Through meticulous observation, he realized that human blood was not all the same. Rather, it contained different types of antigens A and B on red blood cells and corresponding antibodies in the plasma. He identified three main groups: A, B, and O, and later, his colleagues discovered the OB group. Crucially, Landsteiner demonstrated that mixing incompatible blood types led to agglutination clumping of red blood cells, which caused severe, often fatal, transfusion reactions. His discovery meant that for the first time, physicians could match blood types between donor and recipient, ensuring safe transfusion. For this groundbreaking work, Landsteiner was awarded the Nobel Prize in 1930. With the basic science established, The First World War provided the grim proving ground for safe transfusion. Early in the war, transfusions were performed directly from donor to recipient, often a complex and time-consuming procedure in a chaotic battlefield environment. However, the problem of blood clotting during storage still limited its widespread use. The solution emerged through the discovery of anticoagulants. In 1914, Belgian Albert Huston and Argentinian Louis Agot independently demonstrated that adding sodium citrate to blood prevented it from clotting without being toxic, allowing blood to be collected and stored for a short period. This breakthrough was revolutionary. It meant that blood could be collected at a base hospital, stored, and then transported to field hospitals for later use. This laid the fundamental groundwork for the concept of blood banking. The interwar period saw further refinements in blood preservation and storage techniques. But it was the Second World War 1939 to 1945 that truly accelerated the development of modern transfusion medicine on an industrial scale. The massive casualties and widespread trauma of this global conflict created an unprecedented demand for blood and blood products. Recognizing that whole blood was difficult to transport and store long-term, and that plasma, the liquid component of blood, could be used to treat shock and maintain blood volume, large-scale initiatives were launched to collect and process plasma. American and British scientists, notably Edwin Cohn, developed methods for fractionating plasma, isolating components like albumin, which was particularly useful for treating shock. Dried plasma, which could be easily transported and reconstituted with sterile water on the battlefield, became a vital life-saving intervention. As the war progressed, the focus shifted back to whole blood, with further advancements in refrigeration and anticoagulants like the ACD solution, acid citrate dextrose, extending its shelf life. Dedicated blood banks were established, organizing massive donor drives and processing vast quantities of blood. The American Red Cross, for instance, collected millions of units of blood for the war effort. The ability to transfuse blood and plasma rapidly and safely dramatically reduced mortality rates from hemorrhage and shock on the battlefield, allowing more wounded soldiers to survive long enough to receive definitive surgical care. This saved countless lives and fundamentally changed military medicine. The innovations born from the brutal necessity of war had a profound and lasting impact on civilian medicine. Trauma care. The principles of triage, rapid evacuation ambulance services, and specialized trauma surgery developed on the battlefield were directly transferred to civilian emergency medicine, leading to the establishment of trauma centers and improved care for victims of accidents and severe injuries. Surgical advancements. The techniques honed in treating complex war wounds, particularly in orthopedics, plastic surgery, and neurosurgery, found immediate application in civilian hospitals, expanding the scope and success rates of elective and emergency surgeries. Transfusion medicine. The infrastructure, technology, and understanding gained in large-scale blood collection, processing, storage, and safe transfusion protocols during the wars became the foundation of modern civilian blood banking systems. Blood transfusion became a routine and indispensable procedure for a vast array of medical conditions, including major surgeries, severe anemia, cancer treatments, and the management of chronic diseases. The understanding of blood groups further expanded, with the discovery of the Rh factor in 1937 by Landsteiner and Alexander Wiener, which helped prevent hemolytic disease of the newborn and further refined transfusion safety. In conclusion, the crucible of war, while tragic in its human cost, has undeniably been a powerful catalyst for medical progress. From the development of organized trauma care and advanced surgical techniques to the revolutionary advancements in transfusion medicine, driven by the discovery of blood groups and the need for large-scale blood banking, conflict has relentlessly pushed the boundaries of what is medically possible. These innovations, born of military necessity, quickly permeated civilian healthcare, transforming how we treat severe injuries, manage critical illnesses, and save lives in every hospital and emergency room today. The legacy of war in medicine serves as a poignant reminder that even amidst destruction, the human drive to heal and preserve life can lead to extraordinary breakthroughs. For most of human history, the primary threats to life were acute infectious diseases, malnutrition, and trauma. Life expectancy was short, and people often succumbed rapidly to scourges like smallpox, cholera, tuberculosis, or overwhelming infections. The dramatic victories of the 19th and early 20th centuries, the sanitation revolution, widespread vaccination, the advent of antibiotics, and improved nutrition, profoundly altered this epidemiological landscape. As infectious diseases became controllable, and as societies aged, a new dominant set of adversaries emerged: chronic non-communicable diseases. These are long-lasting conditions that often develop slowly, persist over time, and require ongoing management rather than a quick cure. Chief among these, and representing a colossal burden on modern healthcare, are cardiovascular diseases and cancer. The rise of these conditions spurred the development of specialized fields like cardiology and oncology, transforming our understanding of the body's internal dysfunctions and driving a new wave of medical innovation. The heart, a vital pump, has always been recognized as central to life, yet its intricate workings and vulnerabilities remained largely a mystery for centuries. Conditions like dropsy, heart failure with edema, or apoplexy stroke were described, but their underlying causes and specific mechanisms of heart disease were poorly understood. Early physicians had no way to non-invasively assess the heart's function or the state of its vessels. The true birth of modern cardiology began with a series of crucial diagnostic and conceptual breakthroughs in the late 19th and early 20th centuries. The stethoscope and physical examination. Building on Laennec's invention, the stethoscope became an indispensable tool for listening to heart murmurs and abnormal lung sounds that hinted at cardiac dysfunction. Meticulous physical examination, including palpation of pulses and observation of swelling, became more refined. Blood pressure measurement. The development of the sphygmomanometer by Samuel Siegfried Karl von Basch in 1881, and its later refinement by Scipione Rivaroc's and Nikolai Korotkov, provided a crucial objective measure of cardiovascular health. Hypertension, or high blood pressure, was gradually recognized as a silent killer, and a major risk factor for heart attack and stroke. The electrocardiogram ECG. One of the most significant breakthroughs was the invention of the electrocardiogram by Willem Einthoven 1860 to 1927. In 1903, Einthoven realized that the electrical activity of the heart could be recorded. His ingenious device, initially a massive and complex setup, graphically represented the heart's electrical impulses. The EKG allowed physicians to diagnose arrhythmias, irregular heartbeats, heart attacks, myocardial infarctions, and other cardiac abnormalities by analyzing the distinct wave patterns. For the first time, the health of the heart could be objectively assessed in real time, providing invaluable diagnostic information and earning Einthoven the Nobel Prize in 1924. As these diagnostic tools advanced, so did the understanding of the underlying pathology. Atherosclerosis, the hardening and narrowing of arteries due to plaque buildup, was increasingly recognized as the primary cause of heart attacks and strokes. This understanding spurred research into risk factors like high cholesterol, smoking, and sedentary lifestyles. The mid-20th century witnessed the advent of interventional and surgical cardiology. Cardiac catheterization. Werner Forssmann, a German physician, performed the first human cardiac catheterization on himself in 1929, inserting a catheter into his own arm vein and guiding it into his heart. Though initially met with skepticism, his courageous act, which earned him a share of the Nobel Prize in 1956, laid the foundation for diagnostic and interventional procedures like angiography, imaging of blood vessels, and angioplasty, opening narrowed arteries. Open heart surgery. Perhaps the most dramatic surgical frontier was the heart itself. For centuries, touching the beating heart was considered fatal. The development of the heart-lung machine cardiopulmonary bypass in the 1950s by John Gibbon and others was a pivotal moment. This machine temporarily took over the functions of the heart and lungs, allowing surgeons to operate on a still, bloodless heart. This breakthrough enabled complex procedures like coronary artery bypass, grafting CABG to treat blocked arteries, valve repair and replacement, and the correction of congenital heart defects. The first successful open-heart surgery using a heart-lung machine was performed in 1953. Pacemakers and defibrillators. The development of the first implantable pacemaker in 1958 by Arne Larsson with Erik Senning and Rune Elmqvist revolutionized the treatment of slow heart rhythms, while external and later implantable defibrillators offered life-saving interventions for dangerously fast or chaotic heartbeats. These advancements transformed cardiology into a sophisticated specialty, capable of diagnosing, preventing, and treating a wide array of heart and vascular diseases, drastically reducing mortality from the leading cause of death in many developed nations. Simultaneously, another ancient and terrifying disease, cancer, moved to the forefront of medical attention. Cancer, characterized by uncontrolled cell growth, had been observed throughout history. Early treatments were brutal, often involving radical surgery if the tumor was accessible, or little more than palliative care. The understanding of its cellular basis and the development of targeted therapies lagged behind infectious disease breakthroughs. The modern era of oncology began with the understanding that cancer was a disease of cells, a profound shift from the idea of humors or miasmas. Rudolf Virchow's cellular pathology in the mid-19th century laid the groundwork, showing that diseases arose from deranged cells. The identification of cell division and the realization that cancer cells multiply uncontrollably provided the foundational biological understanding. Early cancer treatments were primarily surgical. If a tumor was localized and accessible, surgeons would attempt to remove it completely. Radical mastectomies, developed by William Halsted in the late 19th century for breast cancer, exemplify this aggressive approach, seeking to remove not just the tumor, but also surrounding lymph nodes and muscle, in an attempt to prevent recurrence. The early 20th century saw the emergence of the first non-surgical cancer therapies. Radiation therapy. Following the discovery of X-rays by Röntgen in 1895 and radioactivity by Becquerel and the Curies in the early 1900s, it was quickly realized that radiation could damage and kill rapidly dividing cells, including cancer cells. The first successful use of radiation to treat a cancer patient occurred in 1896. This led to the development of radiotherapy, initially using X-rays and later radioactive isotopes to shrink tumors and prevent recurrence, especially for localized cancers. Chemotherapy early forms. The accidental observation of the toxic effects of mustard gas, a chemical weapon, from WWI on bone marrow and lymphatic tissue, led to the idea that similar chemicals could target rapidly dividing cancer cells. In the 1940s, nitrogen mustard became the first chemotherapy drug used for cancer treatment, initially for lymphoma. This marked the birth of modern chemotherapy, though early drugs were highly toxic and nonspecific, causing severe side effects. The development of new diagnostic tools also advanced oncology. Biopsies, the microscopic examination of tissue samples, became routine for confirming cancer diagnoses and determining tumor type. As imaging techniques like X-rays improved, and later with the advent of CT scans and MRI, physicians could better visualize tumors, assess their size and spread, and guide treatment. The mid-20th century saw the establishment of dedicated cancer research centers and a massive increase in funding, leading to a deeper understanding of cancer's molecular and genetic basis. The realization that cancer was not a single disease, but hundreds of different diseases, each with unique cellular and genetic profiles, transformed research. This led to the development of more targeted therapies, precision medicine, and immunotherapy, which we will explore later. In conclusion, the rise of chronic non-communicable diseases like heart disease and cancer fundamentally reshaped the landscape of medicine in the 20th century. As infectious disease mortality declined, these conditions emerged as the leading causes of morbidity and mortality in aging populations. This necessitated the specialization of fields like cardiology and oncology. Through brilliant innovations in diagnostics EKG, blood pressure, imaging, interventions cardiac catheterization, open heart surgery, and therapeutics radiation, early chemotherapy medicine began to unravel the complex internal dysfunctions of the body. While the battles against these chronic diseases are ongoing and constantly evolving, the foundational work in cardiology and oncology laid the essential groundwork for understanding, managing, and increasingly preventing these complex conditions, moving medicine further into the realm of internal molecular and cellular mechanisms. As medicine transformed from an art into a rigorous science, marked by groundbreaking discoveries and unprecedented interventions, an uncomfortable truth also emerged. Progress, particularly in its early, unchecked forms, often came at a profound ethical cost. For much of history, the patient-physician relationship was largely paternalistic, with the doctor holding unquestioned authority and knowledge. The emphasis on scientific advancement, especially in the late 19th and early 20th centuries, sometimes overshadowed individual human rights and dignity, leading to horrifying abuses in the name of medical research. This period of reflection, sparked by shocking revelations, gave rise to a crucial, indispensable pillar of modern medicine: the formalization of ethical principles, particularly regarding human experimentation and patient rights. Historically, while the Hippocratic Oath provided a moral compass for individual practitioners, first, do no harm, it offered little in the way of a framework for systematic medical research involving human subjects. Early clinical trials were often informal, lacked standardized protocols, and crucially, did not prioritize what we now understand as informed consent. Physicians might experiment with new drugs or procedures on patients without their full understanding or voluntary agreement, driven by the belief that any potential benefit to medical knowledge justified the means. Vulnerable populations, the poor, the institutionalized, minorities, and prisoners were often disproportionately targeted for these experiments, their voices unheard and their rights unrecognized. The most egregious and systematic violations of human rights in the name of medicine occurred during World War II under the Nazi regime. The atrocities committed by Nazi physicians were not isolated incidents, but part of a state-sanctioned program of pseudoscientific experimentation. Prisoners in concentration camps, including Jews, Roma, Poles, Soviet POWs, and disabled individuals, were subjected to unspeakable suffering. Experiments included, but were not limited to, high-altitude experiments. To determine the limits of human endurance at high altitudes for Luftwaffe pilots, prisoners were placed in low-pressure chambers, often leading to agonizing death. Freezing experiments. To study hypothermia for German forces exposed to cold, prisoners were immersed in ice water or left naked in freezing temperatures until they died. Malaria, typhus, and other disease inoculation experiments. Prisoners were deliberately infected with various pathogens to test vaccines or drugs, often without any intention of cure. Sterilization experiments. To advance eugenics, thousands were sterilized forcibly, often through brutal surgical procedures or radiation. Mutilating surgeries and organ removal, performed without anesthesia or medical justification. These experiments were characterized by extreme cruelty, a complete absence of consent, and a callous disregard for human life. When these horrors came to light during the Nuremberg trials after the war, they shocked the conscience of the world. In the Doctors' Trial, 1946 to 1947, 23 Nazi physicians and administrators were tried for war crimes and crimes against humanity. The verdict led directly to the formulation of the Nuremberg Code in 1947. The Nuremberg Code was the first international document to establish ethical principles for human experimentation. Its 10 points laid down foundational rules that are still relevant today. The first and most crucial principle stated, the voluntary consent of the human subject is absolutely essential. This was a radical declaration, asserting that individuals must freely agree to participate in research without coercion and with full knowledge of the nature, duration, and purpose of the experiment, as well as its methods, hazards, and potential effects. Other key tenets included the necessity for experiments to yield fruitful results for the good of society, based on prior animal experimentation, the avoidance of all unnecessary physical and mental suffering, the imperative for the research to avoid death or disabling injury, and the right of the subject to withdraw from the experiment at any time. The Nuremberg Code represented a profound shift, placing the rights and welfare of the individual research participant above the pursuit of scientific knowledge alone. Despite the impact of Nuremberg, ethical abuses continued, particularly in the United States, highlighting that the Code's principles, while groundbreaking, were not immediately and universally adopted or enforced. The most infamous American example is the Tuskegee syphilis study. Conducted by the U.S. Public Health Service from 1932 to 1972, this study involved approximately 600 African American men, most of them poor sharecroppers, in Macon County, Alabama. The stated purpose was to observe the natural history of untreated syphilis. Participants were told they were receiving free treatment for bad blood, but they were never informed of their true diagnosis or the real nature of the study. Crucially, even after penicillin became widely available and proven as an effective cure for syphilis in the 1940s, the researchers actively withheld treatment from these men, preventing them from accessing care, purely to continue observing the progression of the disease. This deception, the denial of life-saving treatment, and the targeting of a vulnerable and marginalized racial group represented a shocking breach of medical ethics and public trust. The Tuskegee study was exposed by a whistleblower in 1972, leading to public outrage, a government inquiry, and ultimately, a landmark lawsuit. Its revelation, alongside other questionable research practices, such as the Willowbrook State School hepatitis experiments on intellectually disabled children, and the injection of live cancer cells into patients at the Jewish Chronic Disease Hospital without their knowledge, galvanized a new wave of ethical reform. In response to these domestic abuses, the U.S. Congress passed the National Research Act in 1974, which created the National Commission for the Protection of Human Subjects of Biomedical and Behavioral Research. This commission produced the Belmont Report in 1979, a foundational document for research ethics in the United States. The Belmont Report articulated three core ethical principles that continue to guide human subjects' research. 1. Respect for persons. This principle emphasizes autonomy, meaning individuals have the right to make their own decisions about participating in research. It mandates informed consent, providing all relevant information and ensuring voluntary participation, and protects vulnerable populations who may have diminished autonomy. 2. Beneficence. This principle requires that research maximize possible benefits and minimize possible harms. It encompasses the obligation to do no harm and to actively promote well-being. 3. Justice. This principle demands fairness in the distribution of research burdens and benefits. It ensures that research subjects are not exploited and that groups who bear the risks of research are also those who stand to benefit from its findings. To ensure these principles are upheld, institutional review boards IRBs in the U.S. or research ethics committees RECs in other countries became mandatory. These independent committees, composed of scientists, ethicists, and community members, review and approve all research involving human subjects, ensuring ethical conduct, participant safety, and valid scientific methodology. No research involving human beings can proceed without their approval. Beyond research, the broader concept of patient rights in clinical care also evolved significantly in the late 20th century. The paternalistic model began to give way to one of shared decision-making and patient autonomy. Key developments include informed consent for treatment. Patients now have the right to be fully informed about their diagnosis, prognosis, treatment options including risks, benefits, and alternatives, and to make voluntary decisions about their care. Privacy and confidentiality. Laws like the Health Insurance Portability and Accountability Act HIPAA in the U.S. 1996 protect sensitive patient health information, giving individuals greater control over who accesses their medical records. Right to refuse treatment. Competent adults have the right to refuse medical treatment, even if it could be life-saving. Advanced directives. Patients can make their wishes regarding future medical care known through living wills or durable powers of attorney for health care. Patient advocacy. The rise of patient advocacy groups has empowered individuals and families to demand more transparent, ethical, and patient-centered care. The evolution of medical ethics is an ongoing dialogue, constantly adapting to new scientific capabilities. Advancements in genetics and genomics, for instance, raise complex questions about genetic privacy, discrimination, and the ethical implications of gene editing. The commercialization of research, global health disparities, and the ethical use of artificial intelligence in medicine present contemporary challenges that require continuous ethical deliberation and the refinement of existing guidelines. In conclusion, the journey of modern medicine has been a testament to human intellect and perseverance, but it has also been a stark reminder of the critical importance of ethical vigilance. The horrifying abuses of human experimentation forced medicine to confront its darkest potential, leading to the establishment of foundational codes like Nuremberg and Helsinki, and principles articulated in the Belmont Report. These, in turn, led to the creation of robust oversight mechanisms like IRBs and a profound shift towards respecting patient autonomy and rights in all aspects of healthcare. The integration of ethics is not an optional add-on to scientific progress. It is an indispensable component, ensuring that the relentless pursuit of knowledge and the alleviation of suffering are always conducted with the utmost respect for human dignity and the inherent value of every individual life. For centuries, medicine grappled with the macroscopic manifestations of disease symptoms, organ dysfunction, microbial infections. While the understanding of anatomy, physiology, and even cellular pathology advanced dramatically, the fundamental blueprints of life, the instructions that guide development, function, and even susceptibility to illness, remained a profound mystery. How were traits inherited? What was the chemical basis of heredity? How did cells know what to do? The answers to these questions lay hidden at the molecular level, in a microscopic helix that would unlock the very code of life and usher in a new era of medical understanding. The age of molecular biology and genetics. The concept of heredity, the passing of traits from parent to offspring, had been observed since antiquity, but its mechanism was unknown. Gregor Mendel's work on pea plants in the mid-19th century laid the mathematical foundation for genetics, introducing concepts like dominant and recessive traits, but he knew nothing of genes or DNA. Early 20th century geneticists, like Thomas Hunt Morgan, confirmed that genes were located on chromosomes within the nucleus of cells. Yet, the chemical identity of the gene, the actual substance that carried this hereditary information, remained hotly debated. Was it protein, widely considered the more complex and diverse molecule? Or was it nucleic acid, the pivotal evidence that DNA was the genetic material came from a series of elegant experiments? In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty demonstrated that DNA was the transforming principle responsible for genetic changes in bacteria. Then, in 1952, Alfred Hershey and Martha Chase, in their famous blender experiment, provided conclusive proof that DNA, not protein, was the genetic material injected by bacteriophages viruses that infect bacteria into their host cells. These experiments decisively pointed to DNA as the molecule of heredity, setting the stage for the next, even more dramatic discovery: its structure. The race to discover the structure of DNA was one of the most exciting intellectual pursuits of the 20th century. At its heart were several key players, each contributing vital pieces to the puzzle. Linus Pauling had proposed a triple helix structure, which proved incorrect. Meanwhile, at King's College London, Rosalind Franklin and Morris Wilkins were using X-ray crystallography to study DNA. Franklin's meticulous X-ray diffraction images, particularly her famous Photo 51, provided crucial data revealing the helical nature of DNA and the spacing of its constituent parts. However, it was James Watson, an American geneticist, and Francis Crick, a British physicist, working at the University of Cambridge, who synthesized all the available evidence and, in 1953, unveiled the correct and revolutionary model: the DNA double helix. Their groundbreaking paper, published in the journal Nature in April 1953, elegantly described DNA as a twisted ladder, with two sugar-phosphate backbones forming the sides and pairs of nitrogenous bases adenine, guanine, cytosine, and thymine forming the rungs. Critically, they showed that adenine A always paired with thymine T, and guanine G always paired with cytosine C, the famous base pairing rule. This specific pairing provided the key to understanding how genetic information could be accurately copied during cell division, and how it could carry the instructions for building proteins. The discovery of the double helix was not merely a scientific curiosity. It was a profound revelation with immediate and far-reaching implications for medicine. It provided the molecular basis for inheritance, mutation, and ultimately, disease. It revealed that genetic information was stored in the sequence of these bases along the DNA molecule, forming a code that dictated the production of proteins, the workhorses of the cell. Errors in this code, or mutations, could lead to faulty proteins or no protein at all, thus causing genetic diseases. This breakthrough marked the definitive birth of molecular biology, a field that investigates biological processes at the molecular level. It unleashed a torrent of subsequent discoveries that rapidly elucidated how the genetic code is translated into functional proteins. The central dogma of molecular biology. Crick proposed the central dogma, stating that genetic information flows from DNA to RNA to protein. Discovery of messenger RNA, mRNA, and transfer RNA, tRNA. These molecules were identified as intermediaries in the process of gene expression, carrying the genetic instructions from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are assembled. Deciphering the genetic code. In the 1960s, scientists like Marshall Nirenberg and Har Gobind Khorana cracked the genetic code, determining which sequences of 3 DNA bases codons corresponded to which amino acids, the building blocks of proteins. The practical applications of these molecular insights began to emerge rapidly, laying the groundwork for what we now call biotechnology and genomic medicine. Recombinant DNA technology, genetic engineering. In the 1970s, the development of techniques to cut, paste, and insert DNA from one organism into another using restriction enzymes and ligases opened up unprecedented possibilities. This allowed for the mass production of therapeutic proteins like human insulin, replacing animal insulin, which often caused allergic reactions, and human growth hormone in bacteria. A revolution for patients suffering from diabetes and growth disorders. Polymerase chain reaction PCR. Developed by Kary Mullies in 1983, PCR allowed for the rapid and exponential amplification of specific DNA sequences. This revolutionary technique became indispensable for diagnosing infectious diseases, e.g., HIV, later COVID-19, forensic analysis, DNA fingerprinting, and genetic research, allowing scientists to study even minute amounts of DNA. DNA sequencing. The ability to determine the precise order of nucleotides in a DNA molecule became increasingly sophisticated. This technology was crucial for identifying disease-causing mutations and understanding genetic predispositions. The ultimate culmination of these molecular biological advancements was the Human Genome Project (HGP). Launched in 1990 and completed in 2003, this monumental international collaborative effort aimed to map and sequence the entire human genome, all 3 billion base pairs of human DNA. The HGP provided a complete blueprint of human genetic information, a reference sequence against which variations and mutations associated with disease could be compared. The impact of the HGP on medicine has been transformative. Identification of disease genes. It has accelerated the discovery of genes associated with thousands of genetic disorders, from cystic fibrosis and Huntington's disease to various forms of cancer and neurological conditions. Understanding disease mechanisms. By understanding the genetic basis of diseases, researchers can elucidate the molecular pathways involved, identifying potential targets for new drugs and therapies. Pharmacogenomics. The study of how an individual's genes affect their response to drugs. The HGP has paved the way for tailoring drug dosages or selecting specific medications based on a patient's genetic makeup, minimizing side effects and maximizing efficacy. Diagnostic testing. Genetic testing has become a powerful tool for diagnosing inherited diseases, assessing disease risk, and guiding reproductive decisions. Gene therapy. The HGP has spurred the development of gene therapy, aiming to treat genetic diseases by replacing, inactivating, or introducing genes into cells. While still an emerging field, it holds immense promise for conditions like severe combined immunodeficiency (SCID) and certain forms of blindness. In summary, the unlocking of the code of life, the discovery of the DNA double helix, represents one of the most profound intellectual achievements in human history. It fundamentally reshaped our understanding of heredity, disease, and the very essence of life itself. This discovery launched the field of molecular biology, leading to a cascade of innovations like recombinant DNA technology, PCR, and DNA sequencing. The culmination of this era, the Human Genome Project, provided the complete genetic blueprint of humanity, a foundational resource that continues to drive scientific discovery and clinical application. From a time when the fundamental unit of inheritance was a mystery, we have entered an era where we can read, manipulate, and even begin to edit the very instructions that define us, heralding a new frontier in personalized and precision medicine. For centuries, if a vital organ failed, a kidney ravaged by disease, a heart weakened beyond repair, or lungs scarred by chronic illness, the outcome was almost invariably fatal. Physicians could offer symptomatic relief, but there was no way to replace a non-functional organ. The concept of taking an organ from one person and successfully transplanting it into another, allowing it to function and sustain life, seemed like science fiction, a feat beyond the capabilities of human medicine. Yet, in the latter half of the 20th century, what was once an impossible dream became a breathtaking reality, driven by brilliant surgical innovation and, crucially, a profound understanding of the immune system. This revolutionary chapter in medicine, focused on organ transplantation and immunosuppression, literally redefined life and death for millions. Early attempts at transplantation, prior to the 20th century were largely unsuccessful and often disastrous. While skin grafts autografts were known to succeed, transplanting organs from one individual to another almost always failed rapidly. The reason, unknown at the time, was immunological rejection. The recipient's immune system recognized the transplanted organ as foreign and mounted a vigorous attack, destroying the new tissue as it would any invading pathogen. The first rays of hope for organ transplantation came from the meticulous work of Alexis Carrel, 1873 to 1944, a French surgeon and biologist. In the early 20th century, Carrel pioneered techniques for suturing blood vessels together vascular anastomosis, a critical prerequisite for connecting transplanted organs to the recipient's circulatory system. His work, for which he received the Nobel Prize in 1912, laid the essential surgical foundation for transplantation, allowing surgeons to reliably connect arteries and veins. However, even with perfect surgical technique, the problem of immune rejection remained. The mid-20th century saw the pivotal breakthrough in understanding and controlling this rejection. Peter Medawar, 1915 to 1987, a British zoologist, conducted groundbreaking research on skin grafts during World War II, studying why grafts from different individuals were rejected, while identical twin grafts were not. His work, along with that of Frank Macfarlane Burnet, led to the concept of acquired immunological tolerance and the understanding of the major histocompatibility complex MHC, or HLA system in humans, as the key determinants of tissue compatibility. They elucidated that the immune system, particularly T lymphocytes, were the primary mediators of graft rejection. For this work, Medawar and Burnet shared the Nobel Prize in 1960. This fundamental understanding of the immune response was the intellectual key that would unlock successful transplantation. With the surgical techniques in place and a growing understanding of immunology, the stage was set for the first successful organ transplants. Kidney transplantation. The kidney, being a paired organ and relatively accessible, was the first solid organ to be successfully transplanted with long-term survival. The seminal moment occurred on December 23, 1954, at Peter Bent Brigham Hospital in Boston. A team led by Dr. Joseph Murray, 1919 to 2012, performed the first successful human kidney transplant between identical twin brothers, Richard and Ronald Herrick. Since identical twins have identical genetic material, immune rejection was not an issue. The transplant was a complete success, and Richard lived for 8 more years. Murray's achievement, for which he received the Nobel Prize in 1990, proved that human organ transplantation was physiologically possible and paved the way for non-twin transplants. The challenge remained for non-identical recipients, where the immune system would inevitably reject the foreign organ. The solution lay in immunosuppressive drugs. Early attempts with radiation and crude drugs were often toxic and ineffective. The real breakthrough came in the early 1960s with the introduction of azathioprine immurin, an immunosuppressant drug developed by Gertrude Ilian and George Hitchings, Nobel Prize, in 1988. Combined with corticosteroids, azathioprine significantly reduced rejection episodes, allowing for the first successful kidney transplants between non-identical individuals. Liver and heart transplantation. With the success of kidney transplants and the development of better immunosuppression, surgeons turned their attention to other vital organs. Dr. Thomas Starzl, 1926 to 2017, performed the first successful human liver transplant in 1967 in Denver, Colorado. While early results were mixed, Starzl's persistent innovation in surgical technique and immunosuppression eventually made liver transplantation a life-saving reality for patients with end-stage liver disease. Perhaps the most dramatic and widely publicized organ transplant was the first human heart transplant, performed by South African surgeon Christiaan Barnard, 1922 to 2001. On December 3, 1967, the patient, Louis Washkansky, lived for only 18 days due to pneumonia. But the sheer audacity and partial success of the procedure captured global attention. While early heart transplant patients also faced high mortality due to rejection and infection, the procedure quickly refined, becoming more successful in the ensuing decades. The true revolution in immunosuppression, leading to widespread transplant success, came with the discovery of cyclosporin in the early 1970s. Discovered serendipitously from a Norwegian soil fungus, cyclosporin was a game-changer. Unlike previous immunosuppressants, it selectively suppressed T lymphocytes, the key cells responsible for rejection, without globally suppressing the entire immune system as severely. This meant less toxicity and a significantly reduced risk of opportunistic infections compared to earlier drugs. Cyclosporin, along with subsequent generations of immunosuppressants like tacrolimus and sirolimus, transformed transplantation from an experimental procedure into a standard, highly successful therapy for end-stage organ failure. The development of modern organ transplantation and immunosuppression has had a profound impact. Life-saving therapies. Millions of lives have been saved or dramatically extended, offering a second chance to patients with otherwise fatal organ failure. For conditions like end-stage kidney disease, heart failure, and liver cirrhosis, transplantation is often the only curative option. Increased understanding of immunology. The challenges and successes of transplantation have vastly deepened our understanding of the immune system, its complexities, and its role in both protecting the body and causing disease. This knowledge has had ripple effects across fields like cancer immunology and autoimmune disease research. Ethical and societal considerations. Transplantation raised, and continues to raise, complex ethical dilemmas. The concept of brain death became critical for defining organ donation from deceased donors. The allocation of scarce organs, issues of organ trafficking, the moral status of recipients, and the lifelong commitment to immunosuppressive drugs all spark ongoing societal and ethical debates. Tissue engineering and regenerative medicine. The success of transplantation has also spurred research into alternative solutions for organ failure, including the development of artificial organs, xenotransplantation animal to human, and the burgeoning fields of tissue engineering and regenerative medicine. aiming to grow or repair tissues and organs from stem cells. In conclusion, the journey of organ transplantation, from an impossible dream to a routine life-saving procedure, is a powerful testament to the convergence of surgical skill, profound immunological understanding, and pharmaceutical innovation. The pioneering work on vascular suturing, the discovery of ABO blood groups, the elucidation of immune rejection mechanisms, and critically, the development of targeted immunosuppressive drugs like cyclosporin, collectively transformed the landscape of modern medicine. It moved beyond merely treating symptoms or fighting infection to replacing entire failing organs, offering a literal rebirth for patients facing terminal illness. This remarkable achievement, while presenting ongoing ethical challenges, stands as one of the most astonishing triumphs in the evolution of modern medicine, forever altering our perception of the human body's resilience and the boundless potential of medical science. For most of medical history, diagnosing internal maladies was largely a speculative art. Physicians relied on external symptoms, patient descriptions, physical examination, palpation, percussion, auscultation, and, in the most dire circumstances, exploratory surgery or post-mortem autopsy. While the discovery of X-rays in 1895 provided the first non-invasive window into the body, primarily visualizing bones and dense structures, it offered limited insight into soft tissues, complex anatomical cross-sections, or dynamic physiological processes. The true revolution in medical diagnostics, allowing for unprecedented, detailed, and often three-dimensional visualization of the body's hidden architecture, came with the advent of three transformative imaging modalities: computed tomography (CT), magnetic resonance imaging (MRI), and ultrasound. These technologies fundamentally changed the practice of medicine, enabling earlier and more precise diagnoses, guiding interventions, and transforming surgical planning. Ultrasound sonography, ironically, has its roots in technologies developed outside medicine. The principles of using sound waves to detect objects were initially applied in sonar for submarine detection during WWI and industrial flaw detection. The medical application began to emerge in the mid-20th century, particularly in the 1940s and 50s. How it works: ultrasound machines use high-frequency sound waves far beyond human hearing, generated by a transducer. These sound waves travel into the body and reflect off different tissues and organs, returning as echoes. The transducer then detects these echoes, and a computer processes the timing and intensity of the returning sound waves to create real-time images. Early development: Karl Dussek, an Austrian neurologist, is often credited with the first diagnostic medical application of ultrasound in 1942, attempting to visualize brain ventricles. Later in the 1950s, engineers and physicians like Ian Donald in Scotland pioneered its use in obstetrics and gynecology, revolutionizing prenatal care. Impact: ultrasound is particularly invaluable because it is non-ionizing, does not use radiation, safe for repeated use especially in pregnancy, relatively inexpensive, and portable. It provides real-time dynamic images, allowing clinicians to visualize blood flow, organ movement, and fetal development. It became indispensable for obstetrics, monitoring fetal growth, detecting abnormalities, and determining due dates; cardiology, visualizing heart structure and function; echocardiography; abdominal imaging, diagnosing conditions of the liver, gallbladder, kidneys, and other soft tissues; guided procedures, guiding needle biopsies and aspirations in real time, increasing accuracy and safety; computed tomography (CT scanning), also known as computed axial tomography (CAT scanning), marked a profound leap beyond conventional X-rays, providing detailed cross-sectional images of the body. How it works. Invented independently by British engineer Godfrey Hounsfield at EMI Laboratories and South African-American physicist Alan Cormack in the late 1960s and early 1970s. Cormack developed the mathematical algorithms, and Hounsfield built the first practical prototype. They shared the Nobel Prize in Medicine in 1979. CT combines X-ray technology with sophisticated computer processing. Instead of a single X-ray image, a CT scanner rotates an X-ray source around the patient, taking multiple images from different angles. A powerful computer then uses these multiple slices to reconstruct detailed cross-sectional or tomographic images of bones, soft tissues, and blood vessels. Impact. The first clinical CT scan was performed in 1971 on a patient with a brain tumor. Its immediate impact on neurology and neurosurgery was revolutionary, as it allowed for the non-invasive visualization of brain tumors, hemorrhages, and strokes with unprecedented clarity, often negating the need for highly invasive procedures like pneumoencephalography. Emergency medicine and trauma. CT quickly became indispensable for rapid diagnosis of internal injuries, e.g., bleeding, organ damage in trauma patients. Oncology. It became a cornerstone for detecting, staging, and monitoring cancer, allowing precise localization of tumors and assessment of treatment response. Musculoskeletal imaging, providing detailed views of complex fractures and joint abnormalities. Surgical planning, offering detailed anatomical maps for complex surgeries. Evolution. Modern CT scanners are incredibly fast, producing images in seconds, with much higher resolution and even 3D reconstructions. While it uses ionizing radiation, the diagnostic benefits often far outweigh the risks, particularly in acute or life-threatening situations. Magnetic resonance imaging (MRI) represents arguably the most sophisticated and powerful non-invasive imaging modality, offering unparalleled detail of soft tissues without using ionizing radiation. How it works. MRI is based on the principles of nuclear magnetic resonance (NMR), a phenomenon discovered in the 1930s by physicists Isidore Rabi, Felix Bloch, and Edward Purcell. Nobel Prizes awarded. Its application to medical imaging was pioneered independently by Paul Lauterbur and Peter Mansfield in the 1970s, who shared the Nobel Prize in Medicine in 2003. MRI works by placing the patient in a powerful magnetic field, which aligns the protons within the water molecules in the body. Radiofrequency waves are then briefly pulsed, knocking these aligned protons out of alignment. When the radio frequency pulse is turned off, the protons relax back into alignment, emitting signals that are detected by the MRI scanner. Different tissues, e.g., fat, muscle, brain, fluid relax at different rates and emit different signals, allowing a computer to create exquisitely detailed cross-sectional images. Impact. The first human MRI scan was performed in 1977. MRI's initial adoption was slower due to its cost, complexity, and scan time, but its unique advantages quickly made it indispensable. Neurology. MRI transformed the diagnosis of brain and spinal cord disorders, including multiple sclerosis, stroke, tumors, and degenerative diseases, providing far superior soft tissue contrast compared to CT. Musculoskeletal imaging, unrivaled for visualizing ligaments, tendons, cartilage, and muscle injuries, e.g., knee and shoulder injuries. Oncology, excellent for detecting and characterizing tumors in various organs, especially those in the brain, breast, and prostate. Cardiology, advanced MRI techniques. Cardiac MRI can assess heart function, blood flow, and myocardial viability. No ionizing radiation. Its major advantage is the absence of X-rays, making it safe for repeated scans and for children and pregnant women. The combined impact of ultrasound, CT, and MRI on modern medicine has been nothing short of revolutionary. They have moved medical diagnosis from inference to direct visualization, transforming virtually every medical specialty. Earlier and more accurate diagnosis. Diseases can be detected at earlier stages, often before symptoms become severe, leading to more effective treatment. Reduced need for exploratory surgery. Many conditions that once required invasive procedures for diagnosis can now be accurately identified non-invasively. Enhanced surgical planning and guidance. Detailed images allow surgeons to meticulously plan complex operations, minimizing risks and improving outcomes. Real-time imaging can guide interventions, monitoring disease progression and treatment response. Physicians can objectively track the size of tumors, the healing of fractures, or the effectiveness of therapies over time. Research. These imaging modalities are indispensable tools for medical research, providing insights into disease mechanisms and normal physiological processes. These technologies, born from physics and engineering, have become indispensable clinical tools. They have empowered physicians with an unparalleled ability to see inside the human body, transforming diagnostics, personalizing treatment plans, and ultimately, vastly improving patient care. The constant evolution of these imaging modalities, from faster scans to more precise functional imaging like fMRI or PET-CT, continues to push the boundaries of what is possible in understanding and treating human disease, truly revolutionizing the way medicine is practiced today. The human immune system is a marvel of biological complexity, a vast and intricate defense network constantly vigilant against an endless array of threats. Bacteria, viruses, fungi, parasites, and even rogue cancer cells. For much of medical history, its inner workings were a mystery, its protective powers recognized only vaguely through phenomena like immunity after surviving an infection, or the success of early vaccinations. However, the 20th century witnessed an explosion of discoveries that began to unravel the profound complexities of immunity, moving from a simplistic understanding of resistance to a sophisticated molecular and cellular mapping of the body's defenses. This deepened understanding not only enhanced our ability to fight infectious diseases and make transplantation possible, but also shed critical light on a paradoxical and increasingly prevalent group of disorders: autoimmune diseases, where the immune system, designed to protect, mistakenly turns its destructive power against the body's own healthy tissues. Early insights into immunity emerged from the successes of vaccination Jenner and Pasteur and the germ theory Koch. Paul Ehrlich's side chain theory in the late 19th century provided an early, conceptual model for how cells might specifically recognize and bind to foreign substances. But the true cellular and molecular basis of immunity remained elusive. The mid-20th century marked a golden age for immunology. Key discoveries included discovery of antibodies immunoglobulins. In the 1930s and 40s, scientists confirmed that specific proteins, later termed antibodies, were produced by the immune system in response to foreign invaders and could neutralize them. Later, the structure of antibodies was elucidated, revealing their Y-shaped form and their precise binding sites. Identification of lymphocytes. The crucial role of white blood cells called lymphocytes, particularly B cells and T cells, as the central players in adaptive immunity, was firmly established. B cells were found to produce antibodies, while T cells were responsible for cell-mediated immunity, directly killing infected cells or coordinating immune responses. Clonal selection theory, proposed by Frank Macfarlane Burnet in 1957, Nobel Prize 1960. This groundbreaking theory explained how the immune system could generate a vast repertoire of specific immune cells, each programmed to recognize a particular antigen. When an antigen is encountered, it selectively activates and causes the proliferation of only those lymphocytes with receptors that fit it, leading to a targeted and highly effective immune response and immunological memory. This provided the theoretical framework for how vaccination worked and how immunity to specific pathogens was established. Major histocompatibility complex, MHC. The discovery of the MHC or HLA in humans in the 1960s, a set of genes that encode proteins on the surface of cells, was critical. These MHC molecules present antigens to T cells, acting like an identification tag, allowing the immune system to distinguish self from non-self. This understanding was, as we've seen, vital for the success of organ transplantation and continues to be central to immunology. These foundational discoveries transformed immunology into a vibrant and rapidly advancing field. They led to improved diagnosis of infectious diseases, serological tests, which detect antibodies against specific pathogens in the blood, became standard diagnostic tools, e.g., for HIV, hepatitis, etc. Enhanced vaccine development. A deeper understanding of the immune response allowed for the rational design of more effective and safer vaccines, moving beyond empirical attenuation to targeted antigen delivery, e.g., subunit vaccines, conjugate vaccines. Cancer immunotherapy, the recognition that the immune system could potentially recognize and eliminate cancer cells, and the later discovery of checkpoint inhibitors that unleash this power, has revolutionized cancer treatment. However, as the immune system's power and precision became clearer, so too did the devastating consequences when it went awry. This led to a growing recognition and understanding of autoimmune diseases. These are conditions in which the body's normally protective immune system mistakenly attacks its own healthy tissues and organs, perceiving them as foreign invaders. The mechanisms of self-tolerance, how the immune system learns not to attack self, are complex, and when these mechanisms fail, autoimmunity results. While some autoimmune conditions, like rheumatoid arthritis, lupus, or type 1 diabetes, had been recognized clinically for centuries, their precise cause was unknown. They were often considered rheumatic or idiopathic, of unknown cause. The advances in immunology provided the tools to finally identify the specific autoantibodies that attack self-antigens and autoreactive T cells that attack self-tissues responsible for these conditions, revealing their true immunological nature. The increasing prevalence of autoimmune diseases in developed countries is a complex phenomenon, likely due to a combination of genetic predispositions and environmental triggers: infections, diet, environmental toxins, hygiene hypothesis, etc. The list of identified autoimmune diseases is extensive and continues to grow, including type 1 diabetes, where the immune system destroys insulin-producing beta cells in the pancreas; rheumatoid arthritis, leading to chronic inflammation and damage in the joints; systemic lupus erythematosus (SLE), a systemic disease that can affect multiple organs, characterized by a wide range of autoantibodies; multiple sclerosis (MS), where the immune system attacks the myelin sheath, protecting nerve fibers in the brain and spinal cord; inflammatory bowel disease, Crohn's disease, and ulcerative colitis; chronic inflammation of the digestive tract; thyroid diseases, Hashimoto's thyroiditis, Graves' disease, affecting thyroid hormone production. The burgeoning understanding of immunology provided not only diagnostic tools, e.g., assays for autoantibodies like ANA for lupus or anti-CCP for rheumatoid arthritis, but also laid the groundwork for novel treatments. Early treatments for autoimmune diseases were largely nonspecific, aimed at broadly suppressing the immune system, e.g., corticosteroids like prednisone, or managing symptoms. While effective in reducing inflammation, these global immunosuppressants came with significant side effects, including increased susceptibility to infections and long-term metabolic complications. However, the late 20th and early 21st centuries have seen a revolution in the treatment of autoimmune diseases, with the development of biologic therapies. These are drugs specifically designed to target key components of the immune response that drive autoimmunity. Unlike traditional immunosuppressants, biologics are typically large protein molecules manufactured using biotechnology. Examples include TNF-alpha inhibitors. Drugs like infliximab and adalimumab target tumor necrosis factor alpha, a key inflammatory cytokine, dramatically improving outcomes for rheumatoid arthritis, Crohn's disease, and psoriasis. B-cell depleting antibodies. Drugs like rituximab selectively target and deplete B cells, reducing antibody production in conditions like rheumatoid arthritis and certain vasculitides. Interleukin inhibitors. Targeting specific interleukins signaling molecules of the immune system involved in inflammatory pathways. These targeted biologic therapies have transformed the management of many autoimmune diseases, offering greater efficacy with fewer systemic side effects, allowing patients to achieve remission and significantly improving their quality of life. The field continues to evolve rapidly, with ongoing research into novel targets and personalized approaches based on a deeper understanding of the specific immune pathways involved in individual patients. In conclusion, the advancements in immunology, driven by foundational discoveries about antibodies, lymphocytes, and self-tolerance, have profoundly reshaped our understanding of health and disease. This intricate knowledge has fortified our defenses against infectious agents and made organ transplantation a reality. Simultaneously, it has cast a powerful light on the enigmatic world of autoimmune diseases, revealing them as conditions where the body's own defense system has gone rogue. The ability to diagnose these conditions with precision, and increasingly, to treat them with targeted biologic therapies, represents a triumph of modern medical science, offering hope and improved quality of life to millions. The continuing journey in immunology promises even more sophisticated interventions, not just for infectious diseases and transplantation, but for the growing challenge of autoimmunity and even cancer, further harnessing the body's own remarkable defense mechanisms. For much of its history, medicine operated on a one-size-fits-all or trial-and-error basis. Treatments were developed and applied broadly, with the expectation that they would work for the majority of patients. While this approach has saved countless lives, it often meant that some patients experienced adverse drug reactions, others found treatments ineffective, and diagnoses were sometimes delayed or imprecise. The reason for this variability, largely unknown for centuries, lies in the unique biological blueprint of each individual. The dramatic breakthroughs in genetics and molecular biology in the late 20th and early 21st centuries have ushered in a revolutionary paradigm shift. Genomics and personalized medicine, where medical decisions are increasingly tailored to an individual's unique genetic makeup and molecular profile. This represents a fundamental reimagining of healthcare, moving towards prevention and treatment that is precise, predictive, and exquisitely customized. The foundation for this revolution was laid by the discovery of the DNA double helix in 1953 and the subsequent elucidation of the genetic code and the central dogma of molecular biology. These discoveries revealed that our hereditary information is encoded in the sequence of DNA, instructing our cells to build the proteins that carry out all life functions. Errors or variations in this genetic code can lead to disease susceptibility or influence how our bodies respond to drugs. The true enabler of personalized medicine, however, was the Human Genome Project (HGP). Completed in 2003, this ambitious international endeavor successfully mapped and sequenced the entire human genome, the complete set of DNA instructions found in each of our cells. This monumental achievement provided the first comprehensive blueprint of human genetic information, a reference sequence against which individual variations could be compared. The HGP dramatically accelerated the development of high-throughput DNA sequencing technologies, making it possible to sequence an individual's genome rapidly and affordably, a feat that was unimaginable just a few decades prior. With the human genome sequenced and the technology to read individual genomes becoming increasingly accessible, the field of genomics was born. Genomics is the study of an organism's entire set of genes, the genome, and how they interact with each other and with the environment. It moves beyond the study of single genes to a holistic view of the entire genetic landscape. This broader perspective has opened up unparalleled opportunities for understanding disease, developing targeted therapies, and tailoring medical care. Personalized medicine, also known as precision medicine, is the clinical application of genomic information. It aims to customize healthcare by considering individual variability in genes, environment, and lifestyle for each person. This approach shifts the focus from a population-based treatment strategy to one that is patient-specific, optimizing efficacy and safety. The impact of genomics on medicine is multifaceted and continues to expand rapidly. 1. Pharmacogenomics. Optimizing drug therapy. One of the earliest and most direct applications of personalized medicine is pharmacogenomics, the study of how an individual's genetic makeup affects their response to drugs. Variations in genes can influence how a person metabolizes a drug, how quickly it's broken down or eliminated, how effectively the drug targets its intended pathway, or whether they are prone to adverse reactions. Example 1. Warfarin dosing. Warfarin is a commonly prescribed anticoagulant blood thinner with a very narrow therapeutic window. Too little is ineffective. Too much can cause dangerous bleeding. Genetic variations in genes like CYP2C9 and VKORC1 affect how individuals metabolize warfarin. By genotyping patients, physicians can more accurately predict the optimal starting dose, reducing the risk of complications and improving treatment efficacy. Example 2. Cancer therapies. Many modern cancer drugs are designed to target specific genetic mutations found in tumors. For instance, the drug imatinib Gleevec revolutionized the treatment of chronic myeloid leukemia, CML, by specifically targeting a fusion protein caused by the Philadelphia chromosome. Similarly, treatments for lung cancer, melanoma, and breast cancer now often involve genetic testing of the tumor to identify specific mutations, e.g., EGFR, BRAF, HER2, that make them susceptible to targeted therapies. This ensures that patients receive treatments that are most likely to work for their specific tumor type, minimizing exposure to ineffective and toxic chemotherapy. 2. Diagnosing and managing genetic diseases genomic sequencing has transformed the diagnosis of rare genetic disorders, many of which were previously undiagnosable, leaving families in frustrating diagnostic odysseys. Newborn screening. Expanded newborn screening panels now include genetic tests for a wider range of inherited metabolic disorders, allowing for early diagnosis and intervention that can prevent severe lifelong disability or death. Preimplantation genetic diagnosis (PGD). For couples at high risk of passing on a genetic disorder, PGD allows for genetic testing of embryos created via IVF before implantation, enabling the selection of healthy embryos. Rare disease diagnosis. Whole exome or whole genome sequencing can rapidly identify the specific genetic mutation responsible for a rare disease, providing a definitive diagnosis, informing prognosis, and sometimes revealing unexpected treatment options. 3. Risk assessment and preventive medicine. Understanding an individual's genetic predispositions allows for more proactive and personalized preventive strategies. Cancer risk. Mutations in genes like BRCA1 and BRCA2 significantly increase the risk of breast and ovarian cancer. Genetic testing can identify individuals at high risk, allowing for enhanced surveillance, e.g., more frequent mammograms and MRIs or preventive measures, e.g., prophylactic surgery. Cardiovascular risk. While complex, genetic variations, when combined with lifestyle factors, can contribute to risk for heart disease or stroke. Genomic information can help tailor lifestyle advice or early interventions. Carrier screening. Genetic tests can identify individuals who are carriers for recessive genetic disorders, e.g., cystic fibrosis, sickle cell anemia, allowing for informed family planning decisions. 4. Infectious disease management. Genomics is increasingly applied to infectious diseases. Pathogen identification. Rapid sequencing of microbial genomes allows for quick and precise identification of pathogens in outbreaks, e.g., during pandemics, tracing their spread, and identifying drug-resistant strains. Guiding treatment. Genomic analysis of bacteria or viruses can identify specific mutations that confer antibiotic or antiviral resistance, guiding the choice of effective medications. The rapid advancements in DNA sequencing technology, leading to a dramatic drop in cost and an increase in speed, are democratizing access to genomic information. However, this revolution also presents significant challenges and ethical considerations. Data interpretation. The sheer volume of genomic data requires sophisticated bioinformatics tools and expert interpretation to distinguish meaningful variations from benign ones. Ethical implications. Issues of genetic privacy, potential for discrimination, e.g., in insurance or employment, the psychological impact of learning about disease predispositions, and the responsible use of genetic information are complex and require ongoing societal dialogue and robust regulatory frameworks. Equity of access. Ensuring that the benefits of personalized medicine are accessible to all, and not just the affluent, is a major challenge for healthcare systems worldwide. In conclusion, genomics and personalized medicine represent a transformative frontier in the evolution of modern medicine. Building directly on the foundational discovery of DNA and the Human Genome Project, the ability to read and interpret an individual's unique genetic code is moving medicine beyond a one-size-fits-all approach to one that is precise, predictive, and tailored. From optimizing drug dosages to guiding targeted cancer therapies, diagnosing rare genetic diseases, and assessing individual health risks, genomics is fundamentally reshaping diagnosis, treatment, and prevention. While challenges remain, the promise of truly personalized healthcare, customized to each person's unique biological blueprint, offers the potential for unprecedented improvements in human health and well-being. Having explored how genomics allows us to read the code of life to personalize medicine, we now turn to an even more audacious frontier: the ability to write or rewrite that code. This is the realm of biotechnology, and more recently and dramatically, gene editing, a scientific capability that has moved from the realm of science fiction into clinical reality, holding immense promise for treating diseases at their fundamental genetic root, while simultaneously raising profound ethical questions about our ability to modify human life. Biotechnology, broadly defined, is the application of biological processes, organisms, or systems to produce products or technologies intended to improve human lives. While ancient practices like brewing and bread making are early forms of biotechnology, the modern era truly began with the advent of recombinant DNA technology in the 1970s. As we discussed, this enabled scientists to cut and paste DNA from different organisms, essentially creating designer DNA. The immediate impact of recombinant DNA technology on medicine was transformative. Recombinant human insulin. Prior to the 1980s, insulin for diabetic patients was extracted from the pancreases of pigs and cows. This animal-derived insulin was expensive, limited in supply, and often caused allergic reactions in patients. In 1982, Eli Lilly introduced Humulin, the first recombinant human insulin produced in E. coli bacteria. This was a monumental breakthrough, providing an unlimited, pure, and inexpensive supply of insulin that was identical to human insulin, revolutionizing diabetes care. Recombinant human growth hormone. Similarly, human growth hormone HGH, previously sourced from human cadavers with risks of viral contamination, could now be mass-produced using recombinant DNA technology, providing safe and effective treatment for children with growth deficiencies. Vaccine production. Recombinant DNA technology also revolutionized vaccine production, allowing for the creation of safer subunit vaccines using only specific proteins from a pathogen or viral vector vaccines, e.g., some COVID-19 vaccines, eliminating the need to grow and inactivate large quantities of dangerous viruses. For example, the hepatitis B vaccine, developed in the 1980s, uses recombinant technology to produce the viral surface antigen in yeast, making it much safer and more scalable than earlier plasma-derived vaccines. These early successes demonstrated the immense power of biotechnology to produce critical therapeutic proteins safely and at scale. This laid the groundwork for the development of monoclonal antibodies, highly specific therapeutic antibodies, gene therapies, and the entire biopharmaceutical industry. However, recombinant DNA technology was primarily about adding or introducing genes. The dream of precisely editing the human genome, correcting faulty genes at their exact location, remained a distant goal. Earlier gene therapy approaches often involved inserting a new, functional gene into a patient's cells using viral vectors. But the insertion point was often random, raising concerns about disrupting other genes or activating oncogenes. What was needed was a tool that could act like a precise molecular scalpel, capable of cutting and pasting DNA at specific, predetermined sites within the vast human genome. This revolutionary molecular scalpel arrived in 2012 with the discovery and development of CRISPR-Cas9, clustered, regularly interspaced, short palindromic repeats CRISPR-associated protein 9. The story of CRISPR is a remarkable example of fundamental scientific discovery, as it originated from curiosity-driven research into the adaptive immune system of bacteria. Scientists, notably Jennifer Doudna and Emmanuel Charpentier, who shared the Nobel Prize in Chemistry in 2020 for their work, along with Feng Zhang and George Church, elucidated how bacteria use CRISPR-Cas systems to defend themselves against viral invaders by cutting and disabling viral DNA. They then demonstrated that this bacterial system could be repurposed as a highly precise, relatively easy to use, and inexpensive gene editing tool in virtually any organism, including human cells. How CRISPR-Cas9 works. At its core, CRISPR-Cas9 consists of two main components. One, a guide RNA-gRNA. This is a small RNA molecule that is engineered to precisely match a specific target DNA sequence in the genome that needs to be edited. It acts like a GPS, guiding the system to the correct location. 2. The Cas9 enzyme. This is a bacterial protein, a nuclease that acts like a pair of molecular scissors. Once guided to the target DNA by the guide RNA, Cas9 makes a precise cut in both strands of the DNA double helix. After the cut is made, the cell's natural DNA repair mechanisms kick in. Scientists can then exploit these repair pathways to disable a gene by introducing errors during the repair process, making the gene non-functional, a gene knockout. Correct a faulty gene by providing a template of the desired DNA sequence, allowing the cell to paste in the corrected version. Insert a new gene by inserting an entirely new piece of DNA at the cut site. The power of CRISPR-Cas9 lies in its unprecedented precision, efficiency, and simplicity compared to earlier gene editing technologies. It has rapidly transformed biological research, allowing scientists to quickly create models of human disease in cells and animals, to understand gene function, and to identify potential therapeutic targets. The clinical applications of CRISPR and other gene editing technologies, such as base editing and prime editing, which offer even more precise modifications without double-strand breaks, are now rapidly moving from the laboratory to human trials, holding immense promise for treating a wide range of diseases. genetic disorders. CRISPR is being investigated for direct correction of mutations causing diseases like sickle cell disease and beta thalassemia. Clinical trials are underway using CRISPR to edit hematopoietic stem cells to correct the genetic defect, offering a potential cure. Cystic fibrosis, correcting the faulty CFTR gene. Huntington's disease and Duchenne muscular dystrophy, silencing or correcting genes causing these debilitating conditions. Cancer immunotherapy. CRISPR is used to engineer T cells, CAR T cells, to make them more effective at recognizing and destroying cancer cells, or to remove genes that suppress the immune response. Infectious diseases. Potential applications include editing human cells to make them resistant to viral infections, e.g., HIV, or directly targeting and disabling viral DNA within infected cells. However, the immense power of gene editing also brings with it profound ethical dilemmas and societal considerations. Off-target edits. Despite its precision, CRISPR can occasionally make unintended cuts at other locations in the genome, leading to potential unforeseen consequences. Germline editing versus somatic editing. Somatic cell editing, modifying genes in non-reproductive cells. Changes are not inherited by offspring. Most current clinical trials focus on somatic cell editing. Germline editing, modifying genes in reproductive cells, sperm, egg, or early embryos. These changes would be heritable, meaning they would be passed down to future generations. This raises significant ethical concerns about designer babies, unintended consequences for the human gene pool, and issues of equity and access. Most international scientific and ethical bodies currently advocate for a moratorium on germline editing for clinical purposes. Equity and access. Who will have access to these potentially curative, but likely expensive, therapies? Will they exacerbate existing health disparities? Societal implications. What are the broader implications of altering the human genome? Where do we draw the line between treating disease and enhancement? The scientific community is actively engaged in developing ethical guidelines and robust regulatory frameworks to govern the responsible use of gene editing technologies. In conclusion, biotechnology, from recombinant insulin to the revolutionary CRISPR gene editing tool, represents a fundamental shift in medicine's capabilities. We have moved beyond merely treating symptoms or compensating for disease to the ability to directly manipulate the very genetic instructions that underpin life itself. CRISPR-Cas9, in particular, has democratized gene editing, making it a powerful research tool and a burgeoning therapeutic modality with the potential to cure genetic diseases, enhance cancer therapies, and combat infectious agents. While offering unprecedented hope, this era also demands profound ethical reflection and careful societal guidance, as humanity now possesses the ultimate power to rewrite the code of life itself, requiring wisdom and responsibility to match its immense scientific prowess. As we stand at the precipice of the 21st century, medicine finds itself in the midst of another profound transformation, one driven not by a single breakthrough, but by the relentless convergence of digital technologies. For centuries, healthcare delivery has been largely a physical, localized endeavor. Patients visiting doctors in offices or hospitals, diagnoses based on human observation and limited objective tests, and medical records painstakingly kept on paper. Today, this paradigm is being radically reshaped by the digital health revolution, characterized by the explosive growth of telemedicine, the burgeoning power of artificial intelligence AI, and the strategic harnessing of big data. This interconnected trinity is not merely optimizing existing practices. It is fundamentally altering how healthcare is accessed, delivered, diagnosed, and understood, promising a future of more accessible, efficient, and personalized care. Telemedicine, at its core, is the delivery of healthcare services from a distance using information and communication technologies. While its earliest forms can be traced back to telephone consultations, the advent of the Internet, high-speed broadband, and ubiquitous smart devices has catapulted telemedicine into the mainstream. The COVID-19 pandemic, in particular, acted as an unprecedented accelerant, forcing a rapid, widespread adoption that might otherwise have taken decades. Suddenly, virtual consultations became the norm, allowing patients to connect with their doctors from the safety and convenience of their homes, minimizing exposure risks and easing the burden on overwhelmed healthcare facilities. The impact of telemedicine is multifaceted. First, it dramatically improves access to care, particularly for individuals in rural or underserved areas who previously faced significant geographical barriers to specialists or even primary care physicians. Patients no longer need to travel long distances, take time off work, or arrange childcare for routine appointments. Second, it enhances convenience and efficiency. Virtual visits can reduce wait times, simplify scheduling, and allow for quicker follow-ups. For chronic disease management, regular virtual check-ins can support continuity of care, enabling earlier intervention for deteriorating conditions. Third, telemedicine facilitates remote patient monitoring. Wearable devices and smart sensors can continuously collect vital signs, blood glucose levels, heart rhythms, and other physiological data, transmitting them directly to healthcare providers. This allows doctors to track a patient's health in real time, identify trends, and intervene proactively, preventing hospitalizations for conditions like heart failure, diabetes, or hypertension. Mental health services have also seen a significant expansion through telehealth, making therapy and psychiatric consultations more accessible and destigmatized. While challenges remain, such as ensuring equitable access across the digital divide and refining reimbursement models, telemedicine has irreversibly cemented its place as an indispensable component of modern healthcare delivery. The second transformative pillar of the digital health revolution is artificial intelligence AI. Far from the sentient robots of science fiction, AI in healthcare refers to algorithms and computer systems designed to perform tasks that typically require human intelligence, such as learning, problem-solving, pattern recognition, and decision-making. At the heart of much of modern AI is machine learning, where algorithms learn from data without explicit programming, and deep learning, a subset of machine learning using artificial neural networks inspired by the human brain. AI's applications in medicine are vast and rapidly expanding. One of its most impactful areas is medical imaging and diagnostics. AI algorithms, particularly deep learning models, can be trained on vast datasets of medical images, X-rays, CT scans, MRIs, pathology slides, to identify subtle patterns that may be difficult for the human eye to detect. For example, AI can assist radiologists in identifying early signs of cancer, e.g., in mammograms or lung CTs, detect retinal diseases from eye scans, e.g., diabetic retinopathy, or classify dermatological conditions from skin images with remarkable accuracy. This augments human expertise, reduces diagnostic errors, and improves efficiency, allowing clinicians to focus on complex cases. Beyond imaging, AI is revolutionizing drug discovery and development. The traditional process of bringing a new drug to market is incredibly long, expensive, and often fails. AI can accelerate this by identifying drug targets, analyzing vast biological datasets to pinpoint the proteins or pathways most likely to be involved in a disease, designing novel molecules, generating and optimizing chemical compounds with desired therapeutic properties, predicting drug efficacy and toxicity, simulating how potential drugs will interact with the body, significantly reducing the need for costly and time-consuming laboratory experiments, repurposing existing drugs, identifying existing medications that could be effective for new diseases. In clinical decision support, AI systems can analyze a patient's electronic health record, including symptoms, lab results, genomic data, and medical history, to provide clinicians with evidence-based recommendations for diagnosis, treatment, and prognosis. This acts as an intelligent assistant, helping physicians navigate the ever-growing volume of medical knowledge and personalize care. AI can also predict patient deterioration, identify individuals at high risk for certain conditions, or optimize hospital resource allocation. While AI's integration raises important questions about data privacy, algorithmic bias, and the ultimate responsibility for clinical decisions, its potential to enhance human capabilities and streamline healthcare processes is undeniable. The fuel that powers both telemedicine and artificial intelligence, and indeed modern healthcare research, is big data. In medicine, big data refers to the massive, complex, and rapidly growing volume of health-related information that is being generated and collected. This data comes from diverse sources: electronic health records, EHRs, detailed digital records of patient encounters, diagnoses, treatments, medications, and lab results, genomic and proteomic data, the vast datasets generated from individual genome sequencing, gene expression analysis, and protein profiling, wearable devices and IoT Internet of Things sensors, continuous streams of physiological data from smartwatches, fitness trackers, continuous glucose monitors, and other connected health devices. Medical imaging, high-resolution images from CT, MRI, ultrasound, and pathology slides. Public health surveillance, data on disease outbreaks, vaccination rates, and population-level health trends. Research databases, clinical trial data, scientific publications, and molecular biology databases. The challenge and opportunity lies not just in collecting this data, but in effectively analyzing it to extract meaningful insights. This requires advanced analytical tools, including machine learning and statistical modeling, to identify patterns, correlations, and predictive indicators that would be impossible for humans to discern from raw information. The applications of big data in medicine are transformative. First, in population health management, analyzing large datasets allows public health officials to identify disease trends, predict outbreaks, understand health disparities, and design more effective prevention programs. For instance, analyzing healthcare claims data can reveal patterns of medication adherence or identify populations at risk for chronic diseases. Second, big data is critical for medical research. By analyzing vast patient cohorts, researchers can identify subtle genetic predispositions, discover new drug targets, and evaluate the real-world effectiveness and safety of treatments outside controlled clinical trials. Third, big data is central to personalized medicine. When combined with genomic information, it allows for the development of highly individualized treatment plans, predictive risk models, and proactive interventions tailored to a patient's unique biological and lifestyle profile. The immense value of big data in healthcare is matched by significant challenges, particularly concerning data privacy, security, and ethics. Safeguarding sensitive patient information from breaches is paramount. Ensuring that data collection and use are transparent, consensual, and equitable requires robust regulatory frameworks and continuous vigilance. Concerns about algorithmic bias, where AI systems might perpetuate or even amplify existing health inequities if trained on biased data, also demand careful attention. In conclusion, the digital health revolution, powered by the synergistic advancements in telemedicine, artificial intelligence, and big data, represents a monumental shift in the evolution of modern medicine. Telemedicine is democratizing access and enhancing convenience, bringing healthcare directly to patients wherever they are. AI is augmenting human intelligence, revolutionizing diagnostics, accelerating drug discovery, and refining clinical decision-making. Big data, the fuel for these innovations, is unlocking unprecedented insights into population health, disease mechanisms, and individual responses to treatment. While this revolution presents its own complex challenges, particularly around ethics and equity, its promise to transform healthcare into a more accessible, efficient, precise, and personalized system holds the potential to profoundly improve human health and well-being on a global scale, fundamentally redefining the future of medicine. As we stand today, reflecting on the evolution of modern medicine, we can look back with awe at the monumental progress achieved. From the earliest understanding of anatomy to the eradication of smallpox, from the invention of anesthesia to the sequencing of the human genome, humanity has dramatically extended lifespan, alleviated immense suffering, and conquered diseases that once seemed insurmountable. The triumphs of the past 2 centuries have ushered in an era of unprecedented health and well-being for a significant portion of the global population. Yet, despite these remarkable achievements, medicine faces a new constellation of complex and interconnected global health challenges that will define its trajectory in the coming decades, necessitating a fundamental shift in approach and a relentless commitment to innovation and equity. One of the most immediate and persistent challenges is the ongoing threat of infectious diseases. While antibiotics and vaccines have curtailed many, new pathogens constantly emerge, and old ones re-emerge with terrifying virulence. The COVID-19 pandemic served as a stark and painful reminder of humanity's enduring vulnerability to novel viruses. It exposed weaknesses in global surveillance, preparedness, and equitable access to vital medical resources, from diagnostics and therapeutics to vaccines. Beyond novel threats, diseases like tuberculosis, HIV/AIDS, and malaria continue to exert a devastating toll, particularly in low-income countries, demonstrating that the fight against established pathogens is far from over. Compounding the infectious disease crisis is the silent, insidious threat of antimicrobial resistance, AMR. This is arguably one of the gravest public health crises of our time. The overuse and misuse of antibiotics in human medicine and agriculture have driven bacteria, fungi, viruses, and parasites to evolve resistance, rendering once powerful drugs ineffective. We are rapidly approaching a post-antibiotic era where common infections and minor injuries could once again become deadly. Surgical procedures, organ transplants, and cancer therapies, which rely heavily on antibiotics to prevent infection, would become prohibitively risky. The challenge of AMR demands urgent global action, including stricter antibiotic stewardship, accelerated research into new antimicrobials, and novel non-antibiotic strategies to combat infection. Simultaneously, the world is grappling with a profound epidemiological transition. As infectious disease burdens decline and populations age, chronic non-communicable diseases, NCDs, have become the leading causes of morbidity and mortality globally. Cardiovascular diseases, cancers, diabetes, and chronic respiratory diseases now account for approximately 70% of all deaths worldwide. These conditions are often driven by complex interactions between genetics, lifestyle factors, such as poor diet, physical inactivity, smoking, and excessive alcohol consumption, and environmental determinants. Addressing NCDs requires a shift from acute care models to long-term chronic disease management, emphasizing prevention, early detection, and personalized interventions throughout a patient's life. Mental health disorders, often neglected and stigmatized, are also increasingly recognized as a major global health challenge, affecting hundreds of millions and contributing significantly to the overall burden of disease. Adding another layer of complexity are health inequities. Despite incredible scientific progress, access to quality health care, life-saving medicines, and preventative interventions remains tragically uneven across the globe and even within countries. Socioeconomic status, geographic location, race, ethnicity, and gender often dictate who receives adequate care and who does not. This translates into vast disparities in health outcomes, life expectancy, and the burden of disease. ensuring global health equity that everyone, everywhere, has the opportunity to achieve their highest possible level of health is not just a moral imperative, but a fundamental prerequisite for global health security, as pandemics and other health threats recognize no borders. Finally, the looming specter of climate change presents an unprecedented health challenge. Rising global temperatures, extreme weather events, changes in precipitation patterns, and air pollution directly impact human health. This includes increased incidence of heat-related illnesses, exacerbation of respiratory and cardiovascular diseases, changes in the geographical distribution of vector-borne diseases, e.g., malaria, dengue fever, food and water insecurity, and the mental health impacts of displacement and environmental disasters. Medicine must increasingly integrate climate science into its public health strategies and clinical practices. In response to these formidable challenges, the future of medicine is poised for revolutionary transformation, characterized by several key shifts. One shift to proactive and preventative care. The focus will increasingly move from reacting to illness to proactively maintaining health and preventing disease. This involves precision public health interventions tailored to communities, coupled with personalized lifestyle recommendations derived from genomic and other health data. Wellness will be emphasized over illness, with greater integration of behavioral science, nutrition, and environmental health into mainstream medical practice. 2. Precision and personalized medicine, reimagined. Building on the genomic revolution, personalized medicine will become even more sophisticated. Beyond genomics, we will see the integration of proteomic study of proteins, metabolomic study of metabolites, microbiome analysis, the trillions of microbes living on and in us, and exposomics, total environmental exposures. This multiomics approach will create a comprehensive, dynamic molecular profile of each individual, allowing for exquisitely tailored diagnostics, disease risk prediction, targeted drug development, and truly individualized therapeutic strategies across all diseases, from cancer to autoimmune disorders. 3. Digital health expansion and integration. The digital health revolution, already in motion, will deepen its penetration. Telemedicine will evolve into ubiquitous, integrated virtual care platforms. Artificial intelligence (AI) will move beyond diagnostic assistance to become a fundamental tool in every aspect of medicine. AI-powered drug discovery, dramatically accelerating the identification of new therapeutics, including novel antibiotics. Predictive analytics, identifying individuals at highest risk for disease progression or adverse events, enabling proactive intervention. Personalized health coaching, AI-driven platforms providing tailored advice on diet, exercise, and mental well-being. Robotics in surgery, enhancing precision and minimally invasive procedures. Digital twins, creating virtual replicas of patients to simulate disease progression and test treatment outcomes. Big data will be ethically harnessed to identify complex disease patterns, evaluate treatment effectiveness at population scale, and inform health policy. 4. Novel and regenerative therapies. The cutting edge of molecular biology will yield unprecedented therapeutic options. Gene editing, CRISPR and beyond, moving from experimental to potentially curative for a growing number of genetic disorders like sickle cell anemia, cystic fibrosis, and various cancers, as precision and safety improve. This also holds promise for creating individuals resistant to certain infectious diseases. Cell therapies. Beyond CAR-T cell therapy for cancer, engineered stem cells and other cell-based therapies will increasingly be used to regenerate damaged tissues and organs, offering alternatives to traditional transplantation. mRNA technology. Building on the success of COVID-19 vaccines, mRNA platforms will be explored for vaccines against other infectious diseases, cancer immunotherapies, and even treatments for genetic disorders by delivering instructions for producing therapeutic proteins. 5. A global and One Health approach. Future medicine will increasingly recognize that human health is inextricably linked to animal health and environmental health. The One Health approach, emphasizing collaborative, multisectoral, and transdisciplinary efforts, will become paramount for addressing zoonotic diseases, those transmitted from animals to humans, food safety, antimicrobial resistance, and the health impacts of climate change. International collaboration and robust global health governance structures will be essential for managing pandemics, sharing scientific advancements, and ensuring equitable distribution of health resources. The future of medicine is not merely about more advanced technologies. It is about a more holistic, integrated, and equitable approach to health. It is about moving from simply extending lifespan to enhancing health span, the period of life spent in good health and free from debilitating disease. It demands continuous ethical reflection, ensuring that scientific power is always wielded with compassion, justice, and a deep respect for human dignity. As medicine continues its timeless quest to understand and alleviate suffering, it will be defined by its ability to adapt to new challenges, integrate revolutionary technologies, and, crucially, extend the promise of health and well-being to every individual on the planet.